Hello, sir. My screen is visible to you?
Hello. Hello, you can start.
Yes, sir. Yes, sir.
You
Visible,
can
visible.
start.
Okay, sir. Thank you. First of all, I'd like to thank the organizer, committee members, all the respected professor, and my co-research fellows for giving me the best opportunity for presenting my work in front of Genius Minds. Hello, my name is Dhruv Joshi. I am studying in master's last sem as a research student in Gujarat. So, cutting trees for the cellulose is being too harsh for the environment. So, as alternative, we, human and scientists, found the best biopolymer, which is produced by bacteria named biocellulose. It has the flexible applications, advanced properties than any other biopolymer, and it is exopolysaccharide with discrete properties like biocompatibility, great mechanical strength, extraordinary absorbency in the hydrated state, elasticity, and many more. So, but the bacteria which gives higher yield of biocellulose is presently limited. So my study, isolation and identification of potent biocellulose producing bacteria from fresh fruits and study of its application in various fields, work to find, identify, screen, isolate, and make the biocellulose for highly potential strain. So there are 12 different fruits and I get 15 bacterial isolates and 6 isolates show the higher pellicle thickness. And these 6 isolates were tentatively identified based on morphological, cultural, and biochemical characteristics. The first 4 was Bacillus species, one is Gluconoacidobacter species, and other one is Rhodococcus species. With the specific substrate and static fermentative procedure, we produce biocellulose in the higher yield. So there will be many applications of biocellulose like food, biomedical, textiles, and wound dressing, and many more, which highlight the flexibility of the material. So this paper exploits the biocellulose production with the help of potential bacterial strain and study of its structural properties, physiochemical nature, and applications and its uses. So bacterial cellulose is one of the much interesting topic and polymer because of their characteristics features like ultrafine fibrils, biodegradability, high water holding capacity, high tensile strength, and not even elasticity and biocompatibility, but also it has a non-toxic in nature, which means this is edible. So there were many species which are used for the starting phase of biocellulose production, but then after the potent bacterial producers like Comagate acetobacter, Gluconacidobacter, the first Acetobacteriaceae family, which founded to be producing biocellulose, as well as the Bacillus, Leishmania, Salmonella, Enterobacter, Pseudomonas, and Shigella in the non-Acetobacteriaceae has high yield of production. The biocellulose have a 3D structure and having the ribbon-shaped fibrils, which have the unit same as plant cellulose is C6H10O5, and their structure were shown in the figure, and this is same as the plant cellulose, but their hydroxyl group have the more improvement than cellulose because of it doesn't contain hemicellulose and other contents which make plant cellulose as impure agent. So synthesis of biocellulose is precisely and specifically regulated multi-step process involving a large number of both individual enzyme and complexes of catalytic and regulatory proteins. I selected the fruit because it has more availability and lower cost for the production, isolation, and many much thing. And there were many shops or many industries which use only fresh fruits, and their pulp will be discarded or dumped. So the dumped or discarded fruit pulps were used for the bacterial cellulose production, which make the better from the waste. So there will be many applications. As I told you that medicinal food made from biocellulose, it means the sugar free, which were used for the patient of diabetes, the Filipino dessert nata de coco, and wound dressing, cosmetic, material for absorbing toxin, ultra fibers, vinegar production, and many, many more. So there were fruits sample, 12 fruit sample, which I collected from the local market of Himmatnagar, Gujarat, my hometown, and the different culture mediums, like the main culture medium, the first bacterial cellulose founded by the Hastings creme medium. Another one is GYE media containing CaCO3, and the further secondary screening, the CAR medium were used. So here are the compositions. For the isolation procedure, I cut the fruits in 20 in the amount of 25 grams. Someone's mic is disturbing me. Hello.
जी बोलिए
Please turn off your mic so I will start again. Thank you. That 25 grams of each fruit was homogenized in the 25 ml of sterile peptone water and mix in the stomacher bag for 1 minute, and their appropriate dilution were spreaded as well as poured in HS agar plate and incubated at 30 degrees Celsius for 24 to 48 hours. But we have the always a backup plan. In this, another method was performed. In 100 gram of fruit sample were blended with 100 ml of distilled water juice, and their 1 ml of juice were transferred to 9 ml HS broth and incubated. In the 2 days of incubation, it showed the pellicles, and the pellicle were used for streaking on the HS agar plate for further isolation process. In the screening, I used GYE agar supplemented with CaCO3, and that isolated colonies were spreaded on that. For the it's the primary screening stage. In that, hydrogel tests were also used. Hydrogel test is based on the Gallardo scale of thickness as poor, fair, good, and excellent, which measure the thickness of pellicles. For the secondary screening, isolates were tried when primary screening were done. But for the excellence isolate, we use secondary screening. As in HS agar plate containing fluorescent brightener dye, I used rhodamine for this procedure, and the colonies which have the bacterial cellulose in their cell wall as an exopolysaccharide, they were bind to that dye, and that will show a fluorescence when observed under the UV lights. And the CAR medium, the potential strain selection method was done by the CAR method, and it showed the color changes. For the identification, I use morphological characteristics, cultural characteristics, biochemical characteristics. As shown in the result, I found the 11 fruits and one other vegetable for this, and their isolates were shown in the table. Their morphological characteristics observation, I performed gram stain, endospore stain, flagella stain, and capsule stain, which were shown in the plus and minus sign. Plus means the containing, minus which not contain. Their cultural characteristics, they're based on their shape, size, margin, elevation, texture, pigment, opacity, and their biochemical characteristics, which the different tastes like catalase, oxidase, indole, methyl red, Vogus proscar, urease, citrate utilization, decarboxylation, and many more, which is the main differential taste were shown in the tables, and other biochemical tests were performed, and their results will be noted. So, for the first 4 bacillus species, I show the morphological characteristics. First image, which shown the endospore staining, and in the second one, which is compared with the Burgess manual of the systematic bacteriology, which show the correct formation and matching of my isolates, which is identified as four bacillus species, and their colony morphology with the excellent match is shown in the second figure. It shows the basic and must containable that I have founded the specific bacillus species. Another species was glucanoacidobacter species, which is shown in the figure. And
Kindly conclude.
okay, sir.
Dhruv Joshi, please conclude.
Okay, sir. So, in the conclusion, I found it that there, from the 12 different fruits, I found 15 strain of bacteria. 6 strain were found to be potential biocellulose producer, identified as 4 species of Bacillus, 1 glucanoacidobacter species, and 1 Rhodococcus species. The market potential of the biocellulose produced was further studied in the field of cosmetic food and biomedical sciences, which shown in the above figure. Thank you. Thank you so much.
Thank you, Dhruv Joshi. Next participant,
Thank you, sir.
Prathamesh.
Excuse me, sir. This is Mohammed Sajjad. May I present? I was first, but somehow I was late.
Okay, ठीक है. You can start now.
Okay, thank you. Thank you for giving me the chance.
It is requested to all the participants, please kindly mute yourself.
May I share my presentation now?
You can share your slide and start.
Just a minute. Okay. Yeah, my screen is visible.
Yes, you can start.
Okay. First of all, I would like to thank the organizer of this wonderful conference for giving me an opportunity to present a piece of my work at this platform. So today's topic is a small piece of work from my research work. That is the anti-candida activity of selected essential oils alone or in combination with the fluconazole and amphotericin B against the drug-resistant isolates of Candida albicans. So as we all of know that there are a lot of fungal infections ranging from mucosal to systemic. They are caused by opportunistic pathogen Candida species, and they are happen to cause diseases like oral candidiasis and vaginitis and candidemia. So to treat these diseases, we have a very good antibiotic, that is a Pauline class of antibiotics, amphotericin B. But this antibiotic has toxicity issues in the host. So the azoles, mainly fluconazole and itraconazole, ketoconazole, they are preferred as a safer drugs for such kind of systemic fungal infections. But the longer and prolonged use of these drugs in the treatment of oral candidiasis, especially in case of immunocompromised patients, has resulted in the development of drug resistance. So we are facing the problem with the antifungal drugs, that is the toxicity issues and the development of drug resistance, especially among the Candida albicans. So in this regard, we are looking for some new effective antifungal agents, maybe some plant products coming from the essential oils, since in our ethnomedicinal value, the essential oils, they have been known from ancient time as a very good source of antimicrobial agents, particularly in our country, India, and other developing countries. And these essential oils, they are found to be safe and without side effects. So we can use these essential oils either as a new agent, or we can use in combination with the existing antifungal agent to potentiate their efficacy. Means the synergistic interaction of oils with the drugs to increase the efficacy of the drugs which are currently used in the chemotherapy. So considering this, we have the hope with the essential oils, and in this regard, we screen certain essential oils for their activity against the drug-resistant fungi, especially their combination with the fluconazole and amphotericin B was tested. So the objective was of this study was simply to determine the incidence of drug resistance among the some clinical isolates of Candida albicans and to assess their anti-Candida activity, and also to check for the in vitro interaction of most active oils with the amphotericin B and fluconazole. So we used 6 essential oils like this: Achillea mellifolium, Ceturosa bachirea, Thymus dinensis, Zetaria multifera, Zizifora chinopodiosis, and Zingiber officinale. These are the 6 essential oils they were used. And the antifungal drugs we use amphotericin B, whereas the strains used, one of the reference strain from the ATCP 10231 was used, and 2 clinical isolates were included in the study. And we perform diffusion assays, time scale assays, and the myces were determined, and also the in vitro interaction was determined using the checkerboard method. So regarding the result, as we using the diffusion assays, what we observed that all the strains they were found to be resistant to the fluconazole and amphotericin B, and further, regarding the activity of essential oils against these drug resistant strains, the 3 oils that is T. dinensis and Z. multifera and Z. clanopodiosis. These 3 essential oils they are found to be highly active against the drug resistant strains in terms of zone of inhibition, almost around 18 millimeter of size. And further, we performed time kill assays to evaluate the cidal efficacy of essential oils, and we found that within 24 hours, these 3 oils they were highly cidal compared to the even amphotericin B and fluconazole, whereas these essential oils was cidal up to the 36 hours of time period. So we found that these test oils they were highly cidal, and if any agent is going to be a cidal in nature, it will not allow the development of resistance against it. Means the fluconazole, it has a problem that this drug is a fungistatic drug, but any drug that is going to be fungicidal, it will not allow the development of drug resistance among the pathogens. So with this is a key factors with these essential oils that they can be used against the drug resistance strains. And further, regarding the MICs, we found that the MIC value for amphotericin B and fluconazole were ranging in the range of 64 to 256 microgram per ml. That indicates a higher level of drug resistance in these isolates. And regarding the tested essential oils, they showed MIC in the range of 90 to 400 microgram per ml. And on the basis of this MIC values, we determined the rate of order, and we found that T. dinensis was the most active with the lowest MIC of 550 microgram per ml, followed by Z. multifera and Z. officinale. And likewise, this is the order of essential oils on the basis of their MIC values. And further, we determine the interaction of oils with the and also with the fluconazole using the checkerboard method. And
please conclude.
Yes, please, I'm coming to conclusion. Regarding this essential oil, we found that among all the tested isolates, with the fluconazole, the Z. multifera was highly synergistic by reducing the MICs by 4 to and 8 folds respectively, whereas with the amphotericin B, the T. dinensis was and Z. multifera were highly synergistic. So in conclusion, what we found out of these 6 oils, the oils of T. dinensis and Z. multifera, they were highly effective in terms of their antimicrobial activity alone, and they were also showing the synergistic interaction with the amphotericin B and fluconazole. So we recommend that these oils they can be used as a safer clinical approach, or they can be used to improve the efficacy of existing drug like amphotericin B and fluconazole in synergistic combination. That's it. Thank you. Thank you.
Okay, thank you.
Ajay
Thank-
sir, may I present my presentation?
Okay, you can start
Yes sir.
your presentation.
Okay.
Is my screen visible?
You can share your screen and start.
Sir, is it visible?
Yes, you can start.
Okay. Good morning, everyone. My presentation is on seasonal bacterial diversity in two mangrove-dominated tropical estuaries and their possible influence on organic matter mineralization. We all know that mangroves are considered to be highly productive ecosystems. So this study is based on the hypothesis which was first proposed by Odom in 1968, where he said that the mangroves transport organic matter to the adjacent waters and nearby ecosystems, which increases their productivity. This entire process is generally termed as an outwelling, and it's influenced by tidal inundation, freshwater input, precipitation, temperature, and the aerial extent of mangrove forests. Now we are particularly interested in the role of bacteria in mangroves on carbon outwelling, and we know that the heterotrophic bacteria are the only organisms that can incorporate organic matter and transfer the organic carbon into the classical food chain through the microbial loop, and bacteria regarded as remineralizers of particular organic matter, and also they are efficient assimilators of dissolved organic matter. For our study, we chose two tropical estuaries, which is in Goa. One is Mandovi and Zwari. We chose two locations. One is within the mangrove forest, and the other within the in the adjacent waters. Our sampling frequency was seasonal during the pre-monsoon, monsoon, and post-monsoon periods, and we had a sampling duration of 2 years. We adopted standard methodologies for environmental parameters like temperature, salinity, pH, dissolved oxygen, chlorophyll A, inorganic nutrients, and the total organic carbon. We also adopted standard methodology for DNA extraction of pure cultures, and we used universal primers for PCR amplification, and sequencing was done by Sanger sequencing method. Our results show that the temperature followed a very typical increase during the pre-monsoon, followed by post-monsoon and monsoon during the study period. Our salinity also showed typical values, highest values during the pre-monsoon, followed by post-monsoon and monsoon period, and there were not much differences between the MMMW and ZMZW, and suggesting that there were well mixed mangrove and adjacent waters. Our pH remained mostly stable, 6.95 to 7.69 throughout the year without much variation. And our DO concentrations varied between 4.5 to 8 mg per liter, suggesting well oxygenated waters throughout the sampling period. When we come to chlorophyll A, it was recorded highest during the pre-monsoon, followed by post-monsoon and monsoon period. And the highest concentrations are attributed to lower turbidity and higher penetration of sunlight. Inorganic nutrients like nitrite, nitrate, and ammonia were highest during the monsoon, which is mostly attributed to the flushing of fresh water from the upstream. Similarly, silicate and phosphate also showed the highest concentrations during the monsoon period, and there are some peaks which can be attributed to higher salinity in phosphate. TOC in water was highest during the monsoon, followed by post-monsoon and pre-monsoon period. Elevated concentrations are attributed to land runoffs and degradation of mangrove litter. Similarly, the TOC concentrations in sediments followed a similar trend, highest in the monsoon, lowest least in the pre-monsoon period, and the values were slightly higher in ZM and ZW in comparison to MM and MW. And when we come to bacterial diversity, we had a total of 651 species, and major phyla were Protobacteria, Actinobacteria, and Firmicutes. In terms of bacterial classes, we had 5 classes: Gamma protobacteria, actinobacteria, bacilli, alpha protobacteria, and clostridia. And this is how it is in MM and MW during 3 seasons. And ZM and ZW also we see a percentage, the percentage of occurrence differs across the locations. And the total number of genera was 33, out of which 28 were found in Mondovi, whereas 25 was found in Zwari. We wanted to do a multivariate statistical approach to see the relationship between diversity and environmental parameters. So we did a Pearson's correlation in which we found that TOC and nutrients showed the most significance with with each of the bacterial species. Now we had 10 environmental parameters and 31 species, so we wanted to be more specific. So we adopted the principal component analysis through SPSS software, and we chose the 4 main components. Component 1 was DO, salinity and pH. Component 2 was TOC and nutrients. Component 3 was chlorophyll A, and component 4 was bacterial community. Using these 4 components, we did a multiple regression model, and we chose the dependent variable as each species, and we found that almost all the species show the significance for component 2, which is nutrients and TOC. We also did a one-way analysis of variance to see the distinction between the variables across the study, and we found that all of the environmentals were significant. Okay, we conclude that our study depicted the occurrence of significant seasonal variability through the individual sampling sites, and variations in the organic carbon and nutrients influence the structure of the bacterial population during also through season. And gamma protobacteria and bacilli are the major bacterial classes involved. Future studies on the functional attributes of bacteria could prove useful. So, thank you.
Ajay sir, can I start?
Ashish ji, you can start after 2 minutes.
Okay, thank you.
Hello, Ashish, you can stop here. I will call you later on. First of all, we have our invited speaker. So, Sir, can I talk to Professor A.M. Deshmukh, sir?
ये लोग बिल्कुल जो आपने इंट्रोडक्शन दिया वो दे दीजिए यार.
Yes, sir. I have joined already.
Okay, sir. I want to introduce you. Dr. A.M. Deshmukh, sir, President Microbiology Society India, was a professor and head Department of Microbiology, Dr. Baba Sahab Ambedkar, Marathwada University, Aurangabad, Osmanabad Sub-Center. He has a long experience in microbiology and has large number of publication. And I'm inviting sir to give a talk on. Sir, please, you can start, sir.
Thank you, sir. So today, in the pleasant morning and pleasant attendee in the microbiology, I'm going to share my thoughts on Microbiologist Society India's work contribution and the importance of microbiology in the world. Today, it's my duty to appreciate Dr. Amritesh Shukla and all his coordinators, colleagues in microbiology, team in microbiology, for arranging such a beautiful event on the topic emerging trends in microbiology. And I'm happy to hear there are large number of registrations and many participants out of India have participated, including USA, have participated and present their work in this international conference. It is my duty again to wish all the best to all speakers, presenters, attendees for contribution in the research of microbiology in India. Now I am coming to the Microbiologist Society India, which was established in 1996. We have completed 25 years. Why I started this association? The reason was, if we want to serve India, it is not always essential to go to Delhi, have a slogan and serve India. You can serve India from UP, you can serve India from Maharashtra, you can serve India from Punjab. At your place, at your university, you can arrange such a beautiful research activity, popularization activity, and serve India. So the Microbiologist Society, now I bet you again, I bet you that there is not a single university or a single institute in India where Microbiologist Society have not reached. There may be, but they are rare cases. So we are successful in inspiring the students, promoting the students, promoting the PhD students, promoting the teachers, and promoting the pioneers, old age persons in the microbiology. We have 900 and more life members from India and from abroad. Again, I will tell you what is the strength of Microbiology Society. Here it is an opportunity. I am thankful to Dr. Amritesh Shukla. He has given me an opportunity to make an appeal to all of you. Join your hands with Microbiologist Society. Again, I am making appeal. What is the strength of Microbiologist Society? Students is the strength of Microbiologist Society. We have more than 15,000 students, annual member of Microbiologist Society. I want to make this number as a 1 lakh students of microbiology, biotechnology, biochemistry, botany, zoology, because I don't consider microbiology as a separate from botany, zoology. It is one and the same, life science. We have to work together. And there are more than 900 like members up to the time. In addition to that, we are conducting many and many activities. Why? You see again, there are other science subjects like a computer science, physics, chemistry, statistics, maths, geology, geography, and many others. All these sciences are required for the development of the human being, but life science is required for existence of human being. Meaning is that henceforth, survival of human being on the earth will not be possible without help of microbiology, biotechnology, or life science. Now you see, you take any problem of the world, the microbiology is essential. For example, malnutrition problem, food problem, who can help you? And we are doing, Microbiologist Society is doing work in single cell protein, popularizing single cell protein. Many of my students are going in the schools and giving lecture in local language on single cell protein importance, probiotics importance, HPV, human papilloma virus vaccine is available, but awareness is not there among ladies. So we have taken a task and our students are going, that is our very important mission, that educating school-going students. Last year we did it, next year also we are doing it. In addition to that, we are arranging various lectures. We are giving different awards. Many awards we have given to inspire young generation. One unique activity of Microbiology Society I would like to tell, we are giving best student recognition memento to one student of every department in India. Understand? There are more than 1,000 departments in India related with the microbiology or biotechnology, and we have given 1,000 memento to one best student of the department throughout the India. So we are encouraging, we are inspiring and making appeal to them. We have a WhatsApp group of only best students of India. We are making appeal to them that you go in the schools and educate the students. In addition to that, you see, we are working at all level, including students level or research worker, top research workers of India. Example, we are organizing international conference out of India. Every year, one international conference out of India is organized by Microbiologist Society. We organized an international conference in Dubai, in Maldives, in Mauritius, in Sri Lanka, in Bangladesh, in Bhutan, in Nepal, in Andaman, in USA also. And this year, our conference is proposed at Tashkent, Uzbekistan. Meaning is that we are having connection, we are trying to make connections throughout the world. Again, one more very important thing we are doing, student exchange program. Students from other country we are bringing in India, and students from India are going in other countries, exchange program, for which the training, accommodation and boarding will be free. Only the students or their institute has to bear travel charges. That is again, you see, beauty of Microbiologist Society. In addition to that, we are giving lifetime microbiology devotion award to the old people. There are many peoples in India which are above 65. They have contributed a lot. And I'm happy to note that last year, Dr. Amritesh Shukla has felicitated 4 to 5 old age microbiologists from UP. That was the pleasant moment in the history of Microbiologist Society. We are organizing many conferences, many training programs, many workshops. Again, you see, the goal of the Microbiology Society is not to earn money, but at the same time, we will never give free. Anything given free doesn't have value. So there are charges, but very, very minimum charges for workshop, for training program, or for different quiz. And the many students are taking training in India on bioinformatics, in food microbiology, in QC and QA, in mushroom technology, in single cell protein, in beer technology, and many other topics. Even in the genetics also, we conducted many trainings. So students are educated. We are educating the students. In addition to that, you see, we have student units in various colleges in India. And I'm happy to tell you, in India, there are many teachers in microbiology and biotechnology. They are very enthusiastic and they are establishing student units through which we conducted. Once we conducted a Rangoli exhibition. Rangoli exhibition in the topic given was immunology. Meaning is that students have exhibited Rangolis on the topic of immunology. Then there was an exhibition of food fermented product. More than 10,000 students throughout the India participated in fermented food exhibition. Like that, we are trying to create awareness in India. It is highly essential in India because, again, sustainable and eco-friendly, these two words are important and they are related only with the life science. So, if we want to develop, again, you see, one more topic I would like to tell you, ethanol production to solve the problem of energy. So, bioethanol, biofuel, such topics, we are organizing various lectures. One lecture series we are organizing, Indians speaking from abroad. That is a very unique activity. Indians speaking from abroad. Meaning is that many Indians are doing PhD abroad. They are doing PhD postdoc abroad. They are serving abroad as a research worker, research assistant, and lectures of these persons are organized, arranged, and what they are telling that I'm here in UK, I'm in Singapore, how my journey was, how I got scholarship, how I tried, and they are requesting that you should also try in the similar manner. This lecture series is inspiring to the many students of India. Every year, we are giving theme, yearly theme. Once there was a yearly theme of biofertilizer. Once there was a theme of pure water for better health. Like that, every year, once there was a theme, protect your teeth. Only regarding the teeth, lectures were arranged throughout the year. This year, educate the children. That is our main theme. Educate children in microbiology so that automatically there will be rush in the colleges and universities of the student in microbiology, biotechnology, or life sciences. And you see, again, one more lastly, I would like to tell very important activity of microbiology is that regularly we are advising to the state governments and to the national government. We have given different letters. Our contribution was great during the corona period. We have supplied 1,000 microbiology students in PCR laboratory when government asked us the help for giving us the research workers in PCR laboratory. Because at that time, everyone was afraid for going in the pathological laboratory. But our students, that is a national service, mind it well. Our students were not afraid. Many of them suffered from corona. Even then, they served for nation. We have given different letters to the government, giving regularly different letters and increasing the importance of microbiology. In many states, there is no postgraduate in microbiology. In Jammu and Kashmir, there is no graduation in microbiology. In 7 sister countries, among seven sister states of India, among 7 states, there are only 4 microbiology departments. We have given letter, and now there is a popularization of microbiology and biotechnology throughout the world, throughout the India. So I am requesting all participants today, they are attending the international conference. I am making them appeal. Please come, join your hands for this noble cause, because we want to uplift the human health in India. We want to solve the problem of malnutrition. We want to solve the problem of toxicity of chemical pesticides and chemical fertilizer. We want to finish it, and for such many noble causes, we have to do work. And my many teachers in biotechnology, botany, zoology, and microbiology, they are working for this cause. In the Uttar Pradesh, I am very happy with all teachers and all coordinators. So I wish them all the best and request them to become a life member, to become an annual member, arrange the activities. And one more thing I would like to add here, Microbiologist Society will support you morally and financially. We don't have monetary problem. Only the noble cause should be there. Noble work should be there. And I'm sure within coming 10 years, we'll totally change the scenario of India by working in the life science. Thank you, sir. Thank you, Dr. Amritesh Shukla and Dr. Rajan, for giving me an opportunity to talk in this very well organized international conference on emerging trends in microbiology. Thank you, sir. Thank you again.
Thank you, sir. Thank you so much for your valuable lecture. And it is very actually informative. And we and our student both get benefited with it, definitely. Thank you, sir, for giving your valuable time. Ashish,
Can I start, sir?
Ashish, you can start now.
Yes, sir. Okay, sir. Thank you.
You can start, otherwise we will give the chance to the other participant.
Yes, sir. I can start, sir. I can start. Sir, so my screen was clear?
Yes.
Sir, can I start? And my screen was
You can
clear
start.
Yes. Good morning, sir. My name is Ashish Kumar Gupta, and I am here to present my research work. The topic is antiviral, antifungal, and antibacterial activities of Pseudomonas aeruginosa, isolate PM1 with plant growth promotion activity. First of introduction, what is rhizobacteria? Rhizobacteria are root colonizing bacteria that facilitate the plant growth directly or indirectly. The mechanism of HPR is directly nitrogen fixation, phosphate solubilization, siderophore, and phytohormone production. If we talk about indirect activities, then they produce antibiotic, hydrolytic enzymes, induce systemic resistance, and exopolysaccharides. Material methods and results. First of all, how to isolate rhizobacteria. Rhizobacteria was isolated from rhizosphere of healthy plant by the soil dilution plating technique, and the pure culture was used for tests. First of all, characterization of bacterial isolates by biochemical characteristics by and colony characterization on the nutrient agar medium, phosphate solution by Vico-Vescia, indole acetic acid test, SCN production, siderophore production, H2S production, and ammonia production. Some enzymatic activities was also detected by catalase test, cellulase activity, lipase activity, amylase activity, and protease activity. And biofilm production was also detected. This picture shows the production of siderophore, phosphorus solubilization, ammonia production, H2S production, SCN production, cellulase activity, lipase activity, protease activity, and amylase activity. And this chart shows the positive and negative result of the test. Nitrate positive, citrate utilization positive, indole acetic acid positive, siderophore production positive, and others. How to induce plant for resistance? SL means side leaf, RSL means remote side leaf. Side leaf were treated with rhizobacteria and remote side left alone. And the virus challenge hold the plants. And we see the result after 3 to 5 days by the development of lesions on the leaf. And the percent immunization was calculated by this formula control minus treated upon control into 100. This picture shows the lesion on the control plants on site and remote side leaf, but the treated plant shows healthy leaves on both site and remote side leaves of tobacco. And
Kindly
the challenge
conclude.
with
Ashish, kindly conclude.
okay, thank you. And antifungal activity and antibacterial activity was also tested for the bacteria. This picture shows the antifungal activity, and this picture shows the antibacterial activity. And the seed growth promotion was also tested for the bacteria, and volatile produced by the bacteria shows the activity. And this analysis was also tested for their metabolites, and this graph shows the metabolites of the bacteria, and this some important compound that detected in the GCMS analysis, and this functions of these compounds. And 16S RNA actually sequencing was also done for the bacteria, and the bacteria identifies as Pseudomonas aeruginosa, and close relation done by the phylogenetic tree. And thank you, sir.
Dr. Ompal Singh, I think he is the next speaker. Dr. Ompal Singh.
जी सर जी सर
Okay, you can start now.
Thank you sir for your invitation and presentation of abstract of 1 minute sir.
Start. You can
Role
start.
of medicinal plant species immunity booster against COVID-19 pandemic. Abstract, the investigation was carried out the medicinal plant of Hathras district of Uttar Pradesh, India. Overall, 10 plant species studied and belong to the 10 families have medicinal properties for immunity booster against coronavirus. The plant species used in different types of pharmaceutical companies and preparation of medicine and CADA kit against COVID-19 pandemic, like as Azadirachta indica, Convolvulus pluricolis, Atotoda vesica, Pithania somnifera, Centella ascetica, Osimum sanctum, Pinospora cordifolia, Terminalia arjuna, Oravia diffusa, Asparagus racemosus. The introduction. The Hathras district is located in western part of Uttar Pradesh, India. It is bounded in the east by Etah district and west Mathura district, in north by Aligarh district and south by Agra district. The Hathras district lies between 20.4 degree to 29 degree north latitude and 76 degree to 77 degree east longitude. Over all the geographical area, about 175.6 square kilometer around this area, and about 390 hectare area covered by forest region of Hathras. Hathras district is divided into the Tehsil of Sikandarab, Hathras, Sadabad, Sasni, and 7 blocks. The Hathras district is under the limit and humid and subtropical climate, and 10 medicinal plant species playing important role in immunity booster against coronavirus in population of Hathras district of Uttar Pradesh. The rural and tribal peoples, races throughout the world have developed their own culture. The medicinal knowledge, a large number of wild and cultivated plants being used them for the treatment of coronavirus. Thus, is considerable amount the information of the medicinal plant is available with these people. So, the difference of the attempt, documentation, and traditional knowledge of the medicinal plant used as a immunity booster during the COVID-19 virus pandemic by the population of Hathras district of Uttar Pradesh, India. Result and discussion. The present investigation reported 10 medicinal plant species belong to 10 families from the district Hathras, and each medicinal plant species following information was provided: botanical name and local name, plant family. Plants are used as immunity booster: root, leaves, stem, flowers, and fruits. Provide the mixed with honey once a daily, 1 a week, improve the immunity booster in human body. And overall, the 10 medicinal plant is immunity booster against coronavirus. Number 1,
Kindly conclude.
Azadirachta indica. Okay, sir. Thank you very much, sir.
Can I start, sir?
Can I start my presentation?
Your name?
Rahnuma Khan.
Sir, first is mandate Anil Kumar.
Just wait for 2 minutes. Just wait for 2 minutes. It's Swati Charasia, Anil Kumar.
Yes, sir.
Anil Kumar, you start your presentation.
Okay, sir. Sir, my screen is visible to you?
Yes, visible.
Yes, sir. So my topic on the isolation and molecular identification of vancomycin-resistant enterococci isolated from freshwater fish in Uttar Pradesh. So first of all,
You open your presentation first. Open your presentation first and then you start.
so this is visible. Yes, sir.
Yes, visible. You can start now.
Okay, sir. Now the what is the antimicrobial resistance? So this is the antimicrobial is the leading problem that is globally recognized. Antimicrobials occur when the microbes evolve the mechanism that protect them from the antimicrobial that we used. The infection caused by the antimicrobial resistant bacteria that is very difficult to cure, and the microbe that resistant to multiple antibiotics or microbials are called multi-drug resistance or the superbugs. So now the introduction to the enterococci. The enterococci are the gram-positive organism which often occurs in diplococci. Bacteria generally found in the human intestine tract, female genital tract, often in the environment. Number of species also identified, but the 65 to 80% role of infection to the clinical environment is done by the enterococci. So why is the vancomycin resistant enterococci is a concern? About 30% of all healthcare associated infection is caused by these bacteria, and these bacteria. This is the report from the Center for Disease Control 2019 about the vancomycin resistant enterococci. It shows that the 54,500 estimate case in 2015, which could be 5,400 estimate death in 2017, and the loss of $539 million. And this is the mechanism of resistance in enterococci. There are two types of mechanism. One is the instillation resistant, that is inherently resistant to vancomycin, that is chromosomal mediated, and another one is the acquired resistance, which is plasmid mediated and transferred to other organism, which is imported to the VRE base. So objective of the study is prevalence of vancomycin resistant enterococci from the fish, isolation, and their molecular characterization. So the first materials and method, we collect the sample from the fish markets and enrich in the bile exclusion azide broth containing 6 microgram per ml vancomycin. This is the sample collection. This is a 60 sample we collect from the gills of the fish markets. Enrichment in bile exclusion azide broth, that is a positive for the presumption of the enterococci. Then the isolation was done in the species agar, specific agar, that is a citrate azide agar, which also contains 6 microgram per ml vancomycin. And the reddish colony on the plate will stick on general media like tryptosoy agar. The biochemical test for identification of genus Enterococcus is a gram-positive staining. Then oxidase, which is negative, and the catalase also negative, and the growth in the 6.5% NaCl and the citrate positive one. Then the molecular identification of enterococci. Out of 60 samples, 47 samples were positive for the enterococci. The most among species is Enterococcus caudiflavus, which is a 46.6%, followed by the Enterococcus faecalis, that is 20%, Enterococcus gallinarium, that is the 6.66%, and the faecium is 5%. This is the molecular identification of the Enterococcus caudiflavus, that is faecalis, faecium, and the gallinarium. Now the now the vancomycin resistance gene, we identified that the resistance isolate of vancomycin resistant enterococci only consist of the chromosomal gene, that is the VAN-C1 and the VAN-C2 in the Enterococcus faecalis and faecium, and VAN-C2/3 gene is Enterococcus gallinarum and casually flavus. Notably, no plasmid mediated gene will be identified in, that is the VAN-A or VAN-B gene. The discussion of the part is that our study show the finding that the vancomycin resistance enterococci of its origin only contain chromosomal gene, that is the VAN-C or
Anilji, kindly conclude.
Okay, sir. So our study shows that there is no plasmid-mediated gene in the vancomycin enterococci of fish origin that could not be transferred to other organisms. And this study show that there is no spread of antimicrobial resistance and no harmful effect on the human health. As the population increase, the fish is the cheapest and the most valuable nutrient food. So without any antimicrobial resistance, fish can be consumed by the human, and there is no impact on their human health. Thank you, sir.
Next speaker is
Sir, can I start my presentation?
may I know who is speaking? Who is speaking?
Sir, can I start my presentation? Sir, can I start my presentation?
Your name is not in the list.
Sir, Rahnuma Khan.
Which number you are listed? At what number you are in the list? आप किस नंबर पे हैं लिस्ट में?
एक सेकंड सर जी.
रवि कुमार यादव को
नाम होगा मैंने लिया था रहनुमा खान.
Ravi Kumar Yadav. Ravi Kumar Yadav is there? Okay, you can start. Rahnuma.
Yes, sir.
Yes, sir.
You have oral presentation?
Sir, poster presentation है.
No, poster presentation is not there. You can leave the meeting immediately.
Okay, sir. Oral presentation. Wheat rust disease in Bahraich. Puccinia graminis triticai causes black rust disease in wheat crop. This fungus has been named Puccinia after the name of famous
Kindly
Indian
stop the
scientist.
sharing with me. Don't
What,
share
sir?
the screen.
Okay, sir. Wheat rust disease in Bahraich. Puccinia graminis triticai causes black rust disease in wheat crop. This fungus has been Puccinia after the famous Indian scientist T. Puccinia. The genus Puccinia is represented by about 1800 species, which are cosmopolitan in distribution and infect wheat, barley, oat, and other members of grass family. The mycelium of Puccinia is intercellular, branched, and septate. Each septum is provided with a minute pore. It may mycelium is provided with small knob or single bacteria which enter in the host cells and absorb food materials. The monokaryotic mycelium is found in the and the eukaryotic is found in the cells. And the bacteria has two hosts. Primary host is that is Triticum aestivum, and the secondary host is Barbary, that is Bulbaris vulgaris. The fungus is called macrocyclic and polymorph. In total cycle, plant habits. In plain of India, it is sown through or November, the infection in February without uridospores. These are found on wheat appear as lemon-shaped vertical lesions on the leaves. It's just 86. Cellular dikaryotic stalked and oval spores. It is a three-layered structure. Over outer layers is thick and sculptured, whereas inner layers are thin. Four equatorial thinner areas are present in outer wall also. These are called germ pores. The eudospores get attached and infect the new plant by producing germ tube. Hence, they just act like conidia. Germ tube penetrates the host epidermis and forms intercellular dikaryotic mycelium. They do not have any resting period and can only infect primary host. Let's we talk about teletospores or teletosaurus. The teletospores are formed by the same hyphae which earlier produced eudospores in northern India. In April, when the crop is fully mature and time of harvesting reaches, the mycelium which
Kindly
produced
conclude.
eudospores.
Ranuma Khan, kindly conclude.
Okay, sir. Okay, sir. And my conclusion is overall, stem weight rust is a really big problem that we must find a solution to before it is too late. If we do not find a solution to stop the expansion of Puccinia graminis or at least slow down its expansion, one of the largest producers of wheat will be destroyed. Thank you. Thank you so much.
Ravi Kumar Yadav is there in the meeting. Next is Ms. Shilpa Supekar.
Yes, sir.
Now, ma'am, you can start your presentation.
Thank you, sir.
Uh, Good morning. Am I audible, sir?
Yes.
Okay. And can you see my screen, sir?
Yes.
Okay, thank you, sir. Now the title of my topic is Epidemiological and Demographic Characteristic of Dengue at Tertiary Care Center in Kutch region during the year 2019. Dengue is the most rapidly sorry for interrupt, sir. Dengue is the most rapidly spreading mosquito-borne viral disease of mankind, with 30-fold increase in global incidence of over the last 5 decades. It is a major public health concern throughout the tropic and the subtropical regions of the world. The present conducted with objective to study epidemiological and demographic characteristic of dengue infection during the year 2019 in the Kutch region, Gujarat. The study was carried out from January 2019 to December at the Department of Microbiology, Gujarat Adani Institute of Medical Science, GK General Hospital at Bhuj. The patient having suspected dengue fever was based on standard criteria like presentation of febrile illness of 2 to 7 days duration were included in the studies. A total of 1,509 blood samples were collected during the study period and the serologically tested for the dengue NS1 antigen and IgM antibodies by the ELISA testing method. The result shows out of these 509 blood samples were tested by the ELISA for the NS1 antigen and the IgM antibodies as per the protocols, and the personal demographic and the clinical details of each patient was recorded. Out of that, 761 tested positive for the dengue. That means the 50% cases comes positive. And amongst all the positive cases, 47 were tested positive by the NS1 antigen test, which help in the early diagnosis of dengue. So rise in dengue cases started after the month of October and it falls down by the end of the December. Analysis of this data revealed that the age group, that is 0 to 10 years, had maximum dengue cases, and the male are more infected than the female. And the dengue cases were high in the urban area. Now I'm showing my table. Table number 1 shows the dengue prevalence rate, where we can see that the 50% cases came the positive out of 1509. 7 comes positive. In the table number 2, it shows the dengue NS1 antigen positive cases by the ELISA. So we detect the 47% cases came positive by this dengue NS1 antigen, and in the dengue IgM antibody positive cases by the ELISA, it shows 46% cases came positive by this dengue IgM antibody, whereas this table shows that we are doing the NS1 antigen and the IgM. So both the dengue NS1 and the IgM
Kindly
antibody
conclude,
positive in the 6% cases.
kindly conclude, madam.
Okay. Now, the conclusion, the present study reported that the dengue mainly affect the children's, males, and the urban populations, and the seasonal increase during the monsoon and the post-monsoon months was reported. Effective implementation of the vector control measure through efforts toward the vector breeding sources reduction and with the use of personal prophylactic measures against the mosquito bites will help in the reducing the dengue prevalence in the community. And in the discussion part, so comparative study of the dengue prevalence rate, well, in the present study, the prevalence rate is 50%, and in the Gujarat
Time is
region,
over.
hello?
Your time is over, ma'am.
Okay. Thank you, sir.
Stop sharing your screen.
Yes, sir.
Next speaker, Ishwardin Chaudhary. Next speaker, Priya. Priya, are you there? Stop sharing your screen.
प्रिया तो है प्रिया द्विवेदी नहीं प्रियंका द्विवेदी
Shiving, Dr. Sachin. Dr. Sachin is there? Dr. Sachin?
bacterial hunter live session.
Dr. Sachin is there?
Yeah, record.
Can you hear me, Dr. Sachin? Okay, he's not there, I think.
Next one.
Next speaker. Next presenter. Who is the next presenter? Kindly present.
नाम देख लीजिए, नाम बोल दीजिए.
Naveen Kumar, Deepak Arya, Gyanendra Kumar, Dr. Gyanendra Kumar. Are you there?
Sir, I am there, Humaira Mukadam.
Okay, ma'am. Please, you can present. You can start your presentation.
Yes, sure. Okay, so is my screen visible?
Yes, visible, ma'am. You can start.
Yes, so good afternoon, everyone. I'm Humaira Mukadam from MIT World-Based University, and I'm going to present on my topic that is biosurfactant-mediated synthesis of chitosan nanoparticles using Bacillus species SH3A and their antibacterial studies against E. coli. Okay, so starting with what are surfactants? Surfactants are surface active agent. They are mostly used in formulation of detergents, skincare products, hair care products, as they have many properties. So starting with what are biosurfactants? Biosurfactants are biological surface active compounds, and they are produced by living cells, mainly by microorganisms. So this is the structure of biosurfactant. As you can see, it contains of a hydrophilic end and a hydrophobic end, which makes it dissolve and separate oil from water. And further, the classification of biosurfactant, it is classified into low molecular weight and high molecular weight biosurfactant. Biosurfactant has various properties. It is very specific. It has a pH and temperature tolerance. It is very relatively easy to prepare. Also, it is philic and hydrophobic in nature. It decreases interfacial surface tension, and it is biodegradable, biocompatible, and it has great digestibility. So these are the applications of biosurfactant in industries. You can see it can be used in petroleum industry, cosmetic industry, agriculture, food, mining, environment, and mostly a detergent and pharmaceutical industry. Okay, so as we have seen that biosurfactant is an important multifunctional biomolecule, hence we have made an attempt to enhance its antimicrobial activity through its incorporation in nanomaterial formation. So nanomaterials, we have synthesized polysaccharide-based nanoparticles. They have low toxicity, high biodegradability, and physiological stability. So one such polysaccharide is chitosan. So we have actually synthesized chitosan nanoparticles and we have combined it with biosurfactant. So, and we are going to check its activity against E. coli. So we have known the prevalence of E. coli infection. It is mostly seen in foodborne infections. And recently, there was an article in Times of India which said that E. coli has caused 1.6 lakh deaths in 2019, and it is going to increase. Also, there is a lot of antimicrobial resistance shown by E. coli. So this is the review of my literature. We have seen that we have read that according to literature, we have known that lipopeptide biosurfactant shows antimicrobial activity. Also, chitosan nanoparticles itself shows certain activities. So the research gap that we have identified, that synthetic surfactants, that is, for example, SDS and all of that, they have complex chemical synthesis and they cause environmental pollution. So Yeah, so synthesis of nanoparticles from bio-based surfactants and biopolymers can also fulfill green chemistry principles. So which will lead to great sustainability and eco-friendly approach. Also, yes. Yes, also utilizing living organism gives us much broader access to sustainable feedstocks. Also, we know that E. coli has been showing antimicrobial resistance, so this will be a great alternative for that, for conventional drugs. So yeah, for our objective was to isolate bacteria from different habitat and then check their biosurfactant producing ability, then the production and extraction of biosurfactant, and then to synthesize nanoparticles, and then to evaluate the antimicrobial activity of this complex against E. coli. So this was our methodology, starting with isolation, then screening of biosurfactant, then extraction of biosurfactant, followed by characterization of biosurfactant, then synthesis of chitosan nanoparticles, followed by characterization by UV spectroscopy, and then evaluating its antimicrobial activity. So these are our results. We isolated 6 bacteria from soil samples and 6 bacteria from water samples, and all of them were maintained in LB slants. Further, this is the biosurfactant ability checked of all the 12 microbes by drop collapse and emulsification test. And as you can see, the SH3A bacteria showed highest emulsification capability and also high drop collapse results. So that was only used for further studies. We also checked its stability in different pH and different temperature, and it showed good stability.
I think
yeah.
You conclude.
Yeah. So this was how the biosurfactant was characterized. We also did FTIR analysis of the biosurfactant and then the confirmation of chitosan nanoparticles. Yeah. So the
fast.
Yeah. So this is the antibacterial activity shown by biosurfactant and nanoparticles. These are the characteristic of our bacteria. And finally, we have concluded that biosurfactant and chitosan nanoparticle complex showed a good antimicrobial activity against pathogenic strain E. coli. Yeah. So I would like to thank the organizing committee of International Conference and our and my university for giving me this opportunity to do the research. So yeah, these are my references. Thank you.
Okay, thank you, Mera. Who is the next speaker? Next oral presenter. Who is the next oral presenter? Pratima. Anushri. Anushri, you can start.
Sir, I am audible?
Anushri, you can start.
Good afternoon, everyone. Anushri here.
Okay, you can start.
Okay, let me share the screen first.
You can start your screen and...
Yes, sir. Yes, sir.
Now your presentation is visible.
Okay. Thank you so much. Good afternoon, everyone. Today I got the opportunity to present my topic on UV radiation-induced synthesis of amylase and their photoprotective function in hypersaline cyanobacterium euhalothic. So my research topic is cyanobacteria. As we all know, cyanobacteria is a diverse group of oxygenic phototrophic prokaryotes. They originated about 3 billion years ago, and they have the capacity of photosynthesis mediated oxygen evolution and development of aerobic life on the earth. The main excellent sources, they maintain the ecological integrity and trophic energy dynamics of the earth system. They are the excellent source of secondary metabolites of medicine, agriculture, industry, such as biofertilizers, biofuels, sunscreens, food supplements that are eco-friendly and economically important. The major concern of my research topic is incidence of UV radiations on photosynthetic organisms, that is cyanobacteria. Solar radiations are the primary source of energy for the photosynthetic organisms. Absolute requirement absorption of solar radiations exposes the photosynthetic organisms like cyanobacteria to the ultraviolet UV radiations. As we all know, UV radiations are categorized into three: UVA, UVB, and UVC. The major concern is of the UVB radiation, that is 350 nanometer to from 280 nanometer to 350 nanometer. UV radiations causes damage to the living organisms. That impact of the UV radiations on the cyanobacteria is they impact their morphology, physiology, biochemistry, structure, cellular integrity of the organisms. As we all know, there is damage to the DNA, damage, growth, survival, motility, orientations. The main focus is how to protect these cyanobacteria, these living organisms, from the UV radiations. The major is what they naturally do. The defense mechanisms that are adopted by them are avoidance, migration, mat formation, change in morphology to increase the self-shedding, or second line of defense is production of antioxidant systems. Third line of defense is production of UV absorbing compounds, and fourth is repair mechanism. The major focus of my study is on the third line of defense, that is the production of UV sunscreen compounds. That are of my research topic is microsporin-like amino acids and cytomelanin. Microsporin-like amino acids, these are the low molecular weight hydrophilic compounds, novel UV absorbing compounds that are found in cyanobacteria under the impact of the UV radiations. They act as a photoprotection, as resistant to various abiotic stressors like heat, short wavelength radiation, desiccation. They are capable of dissipating the absorbed radiation as heat without producing any reactive oxygen species. They are exploited in pharmaceutical cosmetic industry due to their antioxidant capacity and UV sunscreen potential. So research work for this. First of all, I have collected the sample, algal sample from run of Gujarat, and then isolation, cultivation, purification was done in the lab. Isolation was done using the standard microbiological techniques, enrichment method, dilution method, streaking method, and then the sample was cultured in PG11 media under the laboratory conditions. The temperature was maintained 24 to 26 degrees centigrade, and light intensity, fluorescent white light was given 18 watt per meter square, and the cycle was of a light and dark condition. The light was given for 16 hours, and for 8 hours dark condition, light was switched off. And for the after when we got the sample that was impure, isolation, cultivation, after that identification was done. Pure sample that was identified was euhalothic hypersaline cyanobacteria, that was done using the 16S rRNA gene sequencing, and cell morphological traits were observed using the standard microbiological taxonomic keys. And here I have presented the images, cell morphology of euhalothic species under light microscope, fluorescent microscope, and scanning electron microscope. There is a phylogenetic tree which gives the evolutionary combination or a relation with other the ancestors. And next, the main focus was to check the impact of UV radiations. For the to give the UV radiation, artificial UV lights were used. Artificial UV lights were used in closed box, and 3 different UV filters were used, which gave the different light. nanometer. In nanometer, first is 295 nanometer. 295 nanometer UVB light, PAR, UVA, and UVB exposure. 320 nanometer cutoff filter, which gives PAR and UVA light. PAR stands for photosynthetically active radiations, and 395 that gives only the PAR light, that is photosynthetically active radiations. Under the exposure of these radiations, these organisms were able to grow the UV absorbing compounds like MAs. That was as we have taken the spectrophotometer chromatogram, HPLC chromatogram, the partially purified MAs were grown. MAs peaks were shown at 6.4 minute and 10.2 minutes. And same here is the spectral analysis that shows the UV absorption maxima at 331 nanometer and 362 nanometer. That is in my UV radiation range, UV range. MA spectra of HPLC, purified MAs in ULO-thick. These show the structural peaks at the mass spectra of 3031, 303.1 of microsporin to glycine at prominent peak with mass 303.1 and ULO-thick 362 showing the peak at 331.2. Yes, sir?
Try to finish it quickly.
Okay. And after that, under the different cutoff filters, these two compounds were found, MAs, that is MAs ULO-thick 362 and microsporin 2 line glycine, and this effect was checked under the E. coli, that completely given the photoprotective function of MAs on E. coli cells under the UV radiation. The conclusion was that cyanobacteria can resist under the damaging effects of UV radiation in their natural habitat by synthesizing the UV absorbing compounds like microsporin like amino acids, which is a great efficiency for UV screening functions. And moreover, these MAs can be more elucidated in the future as a UV sunscreen, which may be exploited in commercial applications or in cosmetic industries. Already MAs are used in commercial sunscreens, and there is a publication. This work is presented and published in the Elsevier. Okay, sir. Thank you.
Thank you. Next is Pratima, madam. Are you there?
Hello.
Hello.
Pratima, ma'am, you can now present.
Yes, sir. I am present. Share the slideshow. दिव्या देखो बलराम को देखोगे
There is a background noise, ma'am.
Yes, sir. Yes, sir. Please, sir. Wait. I am waiting. Yes. सर को बुला ओपन नहीं हो रहा है सर माय स्क्रीन इज विजिबल हेलो हेलो माय स्क्रीन पे तो विजिबल है शेयर कर रहा है नहीं आ रहा ये आया ना ऑप्शन शेयर पे जाओ
Stay here, Please give chance to the next. Rushalis Walk, please, you can, if you want, you can present.
Sir, is screen visible? Hello? Hello, sir.
And your voice is not clear.
Yes, sir. Now is it clear, sir? Hello? Hello, sir.
Hello? You can start.
Yes. Sir, my voice is clear.
Yes, visible, but your presentation is not visible.
Hello? Sir, am I audible? Hello?
Yes, you audible.
Okay. Good morning, sir. Hello. Good morning, sir. My presentation topic is production of naringinase enzyme by aspergillus flavus and its applicability for debittering of fruit juice. Yes. Hello.
Hello, please
Hello, sir.
present.
Okay.
Only
Stop
one
share.
can present at a time. Rushali, you can present now.
Okay.
Rushalis, please start.
Yes, sir. Sir, is screen visible now? Hello?
Visible. You can now start.
Okay. Yes, sir. Good morning, sir. My presentation topic is production of naringinase enzyme by aspergillus flavus and its applicability for debittering of fruit juice. I'm presenting this topic under the theme area of industrial microbiology. Naringinase is the enzyme, which is one of the important enzyme which plays important role in the debittering of the fruit juice. As concerned to the fruit juice industry, near about 44% of the peel waste is there. And as concerned to the citrus fruit industry, citrus peel contain bitterness due to the presence of the naringinin. This is the substrate for the naringinase enzyme, which is bitter one. And naringin is the substrate which is soluble flavonoid. It consists of a glycon naringinin. And this naringinin is not bitter. Naringinase enzyme is one of the hydrolytic enzyme, and when the naringinase is responsible for the lysis of the naringin, at that time there are the two important steps. First of all, it is responsible to hydrolyze the naringin into L-rhamnose and prunin. And second step, in which it hydrolyzes the prunin into the naringinin and D-glucose. And the naringinin is the non-bitter in the test, means it is responsible for the breakdown of the naringin substrate. These are the objectives of our study. Initially, we collected the soil sample from the rhizosphere soil sample from lemon plant. We also collected the lime pills, and we isolated near about 30 fungal isolates, and out of the 30 fungal isolates, we observed that there is the N1 isolate which was responsible to show the maximum naringinase activity. These are the microscopic images of some fungal isolate, and these three are the microscopic images of the N1. We can see the spore formation of the Aspergillus flavus mycelial network. Septate mycelia is there, septate hypi is there, and as concerned to this graph, actually there are the 4 isolates which was responsible to show the naringinase activity, but when we compared it, the N1 isolate was responsible to show the maximum activity, and we performed the naringinase enzyme activity assay by using the Helmut and Pittner assay. Next one, during the production of the naringinase enzyme, we used different substrate which are waste material, and we perform the solid state fermentation process. You can see here we also use the naringin, which is the standard substrate, and the waste material like the rice straw, sugarcane bagasse, groundnut shells. But as compared to these 3, we observed the maximum activity in the sugarcane bagasse. Means this isolate N1 was responsible, means Aspergillus flavus was responsible to show the maximum activity in 48 hours. And we also optimize during the purification, partial purification of this enzyme, we optimize the concentration of the ammonium sulfate. And we observed that when we are taking the 70% of the ammonium sulfate, at that time, the maximum naringinase activity was observed, and that is the 150 unit per ml. After that, N1 was responsible to show the maximum production of naringinase. Therefore, the 18S rRNA sequencing, which is based on the ITS region, was carried out, and this isolate was responsible to show the more than 98% of the sequence similarity with that of the Aspergillus flavus. Therefore, this organism is Aspergillus flavus. Then the optimization study was carried out by using the one factor at a time process. According to this optimization study, the best substrate was the sugarcane bagasses. Best nitrogen source was the peptone, pH 5, inoculum size is 3%, temperature 28 degrees Celsius. Beef extract is also suitable. That is all about the OFAT. After that, by using the statistical analysis, optimization was carried out. According to the Plackett-Burman design, we can see the p-value. On the basis of the p-value, which is less than 0.05, on the basis of that, we selected the 3 factors for the response surface methodology: sugarcane bagasses, inoculum size, and the temperature is there. Now, according to the central composite design, because PBD is only responsible to show the favorable factors. Now, as concerned to the response surface methodology, these optimized values, sugarcane bagasses is the 66.6 gram per liter, inoculum size is 3.34. According to the response surface plot, temperature which is suitable, that is 31.50. And after completion of this, after optimization, the validation of the CCD model was carried out. And sir, you can see here, under the unoptimized condition, the enzyme activity was 112.5 unit per ml. But after completion of the optimization, means after completion of the OFAT, PBD and CCD, central composite design, the enzyme activity was increased. That was reached up to the 160 unit per ml, and in within only the 48 hours. After completion of this, we confirm our result by using the some analytical method. We also performed the HPLC analysis, and we compared our result with the standard. The retention time, which is 13.4, which is near about the standard. And we were also interested in the secondary, we want to study the secondary structure of the protein. We also perform the CD spectroscopy. According to the CD spectroscopy, we diluted the sample 2 times, and the result was it contained the 27% of the alpha helix and it contained the 50% of the beta sheets. And when we compared it with the standard one, at that time also the closeness was observed, similarities was observed. Now, as concerned to the application, naringinase plays important role in the debittering of the fruit juice, because when we are talking about the fruit peels, it contains the bitterness. And if the some content of the peel is present inside the fruit juice, then the bitterness is there, and naringinase enzyme is significantly responsible to decrease the bitterness. And this experiment was carried out by the Davis method. You can see here the different treatments we are using, means we are taking the different concentrations of the enzyme, different time intervals. In our actually, we are checking the naringin content. Now, when we check, just a minute, when we check the naringinin content, at that time the best result which we observed that in 1 hour, hello, when, sir, am I audible? When we are taking the 1 microgram per ml of the naringinase enzyme, then it is responsible for the debitterness. Means initially the bitterness was 44.94, and it is responsible to decrease the bitterness, responsible for the debittering. Then it reached up to the 34.83. In within only the 3 hours, the bitterness get decreased. Actually, as concerned to the conclusion, these isolate aspergillus flavus N1 isolate was responsible for the maximum production of the naringinase enzyme, and before optimization the enzyme activity was low, and obviously after completion of the optimization, means by using the optimized value which is shown by the Minitab software when we perform the CCD, then by using these optimized values during the validation, the enzyme activity get increased, and yes, this enzyme which is produced by the fungal strain N1 Aspergillus flavus, it shows the significant result and plays the important role for the debittering of the fruit juice. And yes, this enzyme should be used in the industry to remove the bitterness from the fruit juice. Thank you, sir.
Okay, I want to stop the oral presentation here because one of our resource person is giving talk over here.
Sir, can I
So
stop
we will
my
resume it later on.
Hello. Sir, can I stop my presentation?
No, after some time you can resume. Amrita ma'am, you can start. Amrita ma'am. Can you hear me? But after first of all, I want to introduce you with our audience. Then then you will start. Please wait for a few minutes. Okay, ma'am.
Dr. Amitaran, postdoctoral researcher at Applied Microbiology and Applied Technology School, the Peninsula University of Technology, that is from IIT Goa, India, and the topic of her thesis was making off from rice straw process development and optimization. An area of research is biomass to molecule, industrial municipal agricultural waste, wastewater bioremediation, valorization microbial biorefineries for production of biofuels and other valuable products, bioremediation, industrial microbiology, fermentation, aromatization, microbial production of bioplastics, human God ASC research to create one-woman scientists, where one-woman researcher scholarship to science, and Erasmus Nobel Scholarship, Karawata Berlin 2004, awarded with full funded postdoctoral scholarship at University of California, Valencia, Spain. Now I'm starting the talk of Dr. Amrita.
Ma'am, now you can start. Amrita ma'am, you can start now.
Hello, good morning, everyone. Hope you can hear me.
Yes, ma'am. Yes, ma'am, you start now. Your slide is visible, ma'am. You can start.
Ajay Srivastava sir, I can see that you are saying something, but unfortunately, I cannot hear you.
Ma'am, your screen is your slide is visible and your voice is clear. Now you can start.
Ma'am, you are audible. You can start now.
Okay. Good morning, everyone. I'm Dr. Amrita Ranjan. I believe you all can listen me. Unfortunately, I cannot listen you all. But as you all can listen me, I think we can forward. We can just continue with this session. So let me share something about you. I'm Dr. Amrita Ranjan. I'm working as a researcher at MB, Cape Peninsula University of Technology in Cape Town. Currently, my research team works on utilization and valorization of various agricultural and industrial waste and to produce some biofuels and value added products from it. We preferably do this through microbial route. And today I will be talking about one of those projects which we are handling currently and which is basically on microbial bioplastic production.
Ma'am, your slide is visible. You can continue.
Okay, sorry, I was just struggling with some technical issues and I hope it's sorted now. So before starting the presentation, we will like to discuss something about the research outline we are going to discuss in this presentation. We will start with knowing about bioplastics. Bioplastic क्या है and how they are compatible or You can compare them with petroplastics, which are also known as synthetic plastic, जो कि हम daily use में आजकल use करते हैं. What is the need for bioplastic in current scenario? And generally, bioplastics are also classified into some groups. We will also get to know about these classification. And then finally, the most important part of it is to know about PHA. PHA is polyhydroxyalkanoates. And these are basically bio-based biopolymers. And we will learn about these PHA. We will also get to know about how the microbial entities produce these PHA. Although PHAs can also be produced from plants, but today we will be discussing about their microbial route. We will also talk about the biosynthesis and degradation of PHA. And finally, we will discuss that what are the different microbes which can produce these PHAs or bioplastics, and what are the challenges we face while producing them. And finally, we will definitely talk about the application of these PHA in the current world. So let's start with bioplastic. बायोप्लास्टिक के बारे में आई एम प्रिटी श्योर बहुत लोगों ने सुना होगा, और अगर बायोप्लास्टिक नहीं सुना तो आपने प्लास्टिक के बारे में तो जरूर सुना होगा, क्योंकि आजकल प्लास्टिक जो है ऐसा कोई एरिया नहीं है जहां आपको प्लास्टिक नहीं मिलता. चाहे आप नैनो टेक्नोलॉजी की बात करें, चाहे आप बड़े बड़े फर्नीचर्स या बिग कंस्ट्रक्शंस की बात करें, प्लास्टिक इज समथिंग विच इज एवरीवेयर. So, but we all know that this plastic is creating lots of havoc for us and is not something which we are actually looking forward to. But इसकी utility इतनी ज़्यादा है and we are so much dependent on it कि हमें ज़रूरत है किसी ऐसे material की जो कि basically biopolymer हो, जिसकी qualities बहुत ज़्यादा similar हो plastic से, but at the same time it should be biodegradable and eco-friendly. So in search of that, the whole world is actually looking forward for a biopolymer, which is very similar to plastic. And we generally call them in general as bioplastics. Although ये जो definition है bioplastic की, ये person to person या researcher to researcher थोड़ी vary करती है. But when we talk about it, we generally talk about it. Bioplastic are the biodegradable material that come from renewable resources. And why we are talking about it? Because these are the potential substitute for synthetic plastics. So these are made from natural and renewable resources, and they help us in giving independence from fossil fuel or petroplastics. We all know that the synthetic plastic which we are using are quite versatile in terms of their strength, their flexibility, and their rigidness. Depending upon our need, we select our synthetic plastic. Similarly, the bioplastics are also trying to be developed in the same fashion. The bioplastics or the PHAs we will discuss, they comes in different varieties, and today in my coming up slides we will discuss them more. The global production of these bioplastic till 2022 by European bioplastics has been reported to be 2.23 million tons, while we target to get it to 6.3 million tons by the year 2027. But believe me, we need more of it. And that's why we all are researching to find an alternative route or to find a suitable microbe that can produce good quality as well as quantity of these bioplastics so that we can exceed our target. Petroplastics versus bioplastics. As हम सबको जनरली इस बारे में काफी आइडिया है, बट स्टिल जस्ट टू गो थ्रू इट, वी नो दैट पेट्रोप्लास्टिक जो है they deteriorate our environment because they are not biodegradable, and depending on their structure, plastic can take from 20 to 500 years or even more. The PET plastic we generally use in our household items, वो सबसे ज्यादा टाइम लेता है डिकम्पोज होने में, sometimes even 1,000 years. And these are the PET plastic specifically, which is a fossil based plastic. They never get degraded by microbes. They generally degrade through sunlight and through extreme environments, like high temperature and these kind of extreme condition. But the problem is when they degrade, they don't degrade in a simple manner. They transform into small fragments, which are also known as microplastics, which creates much more issues and much more problem to environment, because the very small size of the microplastics, because of that, they find a way to enter into our food chain. And it's not only our food chain, they also enter into the food chain of the aquatic life. And that is how they are creating many diseases, as well as they are affecting aquatic life and humans very badly. So as we can see, these are non-biodegradable and non-compostable. We look for a substitute, which is bioplastic. And bioplastics are environment friendly. They are biodegradable polymeric compounds, and they are composed of covalently bonded monomeric units. They are primarily derived from biological sources such as plants, bacteria, microalgae, photosynthetic bacteria, actinomycetes, and so on. And they are quick at degradation. They generally take 3 to 6 months to degrade completely. Now, what is the need for bioplastic? It's a very simple cycle which I have shown here, just to understand that, as we know, we try to utilize renewable resources to produce these microbes, and these microbes will produce PHA. The PHA will go into the industry and will be further converted into various utility products, from cutleries to furnitures to anything you can think of which is made of plastic. And because they are biodegradable, they can be easily decomposed further. And while decomposing, they will release carbon dioxide and water. So they can further undergo the anaerobic digestion. The sludge which remains can be used as a manure or fertilizer, and this manure can further be utilized to grow more plants and their wastes. So this is a cycle that contributes to circular economy, and it helps to maintain the natural cycle of the earth. Now let's talk something about the bioplastics and their classification. This is the classification which was given by European Bioplastic. And the bioplastics, which are generally petroleum based, they are mostly non-biodegradable, or they take too much of time to degrade. And the most common ones which we generally use are polyethylene, polyethylene tetrafactylate, and polypropylene. While today we are going to discuss about bio-based bioplastics, and more specifically about the polyhydroxyalkanoates, and which are produced by the microbes. Although another class of bio-based bioplastic, which is very common and is under research, is PLA, which is generally produced from plants. So plastics can also be classified as the thermoplastics and the thermosets. Thermoplastics are the plastics that can be molded, while the thermosets are the plastic resins which polymerizes and cross-links upon heating and are unmoldable. The examples of thermoplastics from petrochemical feedstock are polyethylene and polypropylene, and PET, the polyethylene tetrafilate, while the common thermosets includes the PF resins, the polyurethanes, and etc. These mentioned plastics can be either replaced or enriched. मतलब या तो आप उनको completely replace कर दीजिए bio based plastic से, या you just add them as a additive or filler, and with these bio based plastics, which is PLA or different kinds of PHA or starch based polymers. So the bio-based plastics can be used in both the ways, as an additive to the petroleum-based plastics or as an independent bioplastics. So if we look into the global production capacity of bioplastic, in 2027, we target 6.3 million ton production. And currently, the bio-based non-degradable plastic, जो कि आप यहाँ लेफ्ट साइड में आप नीचे देख सकते हैं बायो बेस्ड नॉन डिग्रेडेबल प्लास्टिक, जिसका टारगेट है 43.5 अटैच अचीव करने का. Currently, this is at 48.5%. So we need to reduce the use of this from 48 to 43, and we need to increase the production of biodegradable, which is currently 43.5, and we are targeting in 2027 to be as 56.5%. So, और इस बायोडिग्रेडेबल प्लास्टिक का भी जो क्लास विच इज पॉलीहाइड्रोक्सी एल्कोनोइड, जिसके लिए हम स्पेसिफिकली वर्क कर रहे हैं, this is currently at 3. It contributes only to 3.9%, and we want in 2027 it should achieve at least to 8.9 or close to 10%. So this is our target for 2027. And hopefully, with the research going on, we will surely achieve it globally. Now, let's discuss about polyhydroxyalkanoate. Polyhydroxyalkanoate are biopolymers. They are the major components of microbial cell. हम सबने पढ़ा है किसी भी माइक्रोबियल सेल में प्रोटीन होता है, पॉलीसेकेराइड होता है, लिपिड होता है, अमाइनो एसिड्स होता है, और उसी के साथ बहुत सारे माइक्रोबियल सेल्स में पॉलीहाइड्रोक्सी एल्केनोएट भी मिलता है. ये जनरली इंक्लूजन्स के फॉर्म में मिलता है, जिसे हम पीएचए ग्रेन्यूल्स भी कहते हैं. इनको हम कार्बोनोसोम्स के नाम से भी जानते हैं. These inclusions are generally cytoplasmic inclusion, जो साइटोप्लाज्म में ग्रेन्यूल के फॉर्म में प्रेजेंट होते हैं. ये कोई भी सेल, कोई भी माइक्रोबियल सेल पीएचए क्यों बनाएगा? क्योंकि जब it's a very simple fundamental thing that it is a way to store energy, but to use this energy when the cell actually needs it. जब तक मीडिया में सरप्लस अमाउंट ऑफ न्यूट्रिएंट रहेगा और गुड कंडीशंस या ऑप्टिमल कंडीशंस रहेंगी उस माइक्रोब के ग्रोथ के लिए, तब तक वो थोड़ा अपना पार्ट स्टोर करेगा ग्रैन्यूल्स में, लिपिड ग्रैन्यूल के फॉर्म में. वंस द कंडीशंस गेट एडवर्स और द न्यूट्रिएंट्स इन द मीडिया ये डिप्लीट होने लगेंगे, तो अपने सर्वाइवल के लिए वो ये जो उसने स्टोरेज ग्रैन्यूल बनाया है, इसमें जो लिपिड है, इसको वो यूटिलाइज करेगा ताकि वो जिंदा रहे विद द होप कि शायद उसको ऑप्टिमल कंडीशंस जल्दी मिल जाएंगी। ये केवल माइक्रोब पे नहीं, ये चीज हर लिविंग ऑर्गेनिज्म पे अप्लाई होती है। जस्ट लाइक कि जब तक हमारी सैलरी अच्छी आती है, वी ट्राई टू सेव समथिंग फॉर आवर डिफिकल्ट डेज so that we can survive during difficult days. So in the similar fashion, microbes also do it. They save some of their they store some of their energy in the form of these granules, so that if ever any adverse climatic condition or the nutritional condition comes, then they can survive for some time. But definitely, this is not characteristics of all. Only certain microbes can do this. So now the point is ki, what are these optimal conditions? Optimal condition किसी भी माइक्रोब के लिए क्या होगी कि उसको खाना अच्छे से मिले, जो उसकी ग्रोथ के लिए इम्पोर्टेंट है वो मिले, उसमें सो दैट कि जो डीएनए एंड आरएनए उसको बनना है अपने मल्टीप्लिकेशन के लिए, प्रोटीन सिंथेसिस के लिए, वो सब प्रॉपर बने। एंड जो फिजिकल कंडीशंस हैं जैसे कि टेम्परेचर, ऑक्सीजन, पीएच, ये भी ऑप्टिमम रहे उसकी ग्रोथ के हिसाब से। बट अगर हम टारगेट कर रहे हैं कि हमें बैक्टीरिया में पीएचए प्रोडक्शन चाहिए, तो हम उसको पूरी ऑप्टिमल कंडीशंस बिल्कुल नहीं दे सकते, क्योंकि अगर हम उसको कंप्लीट ऑप्टिमल कंडीशन दे देंगे तो जो बैक्टीरिया है वो नॉर्मली ग्रो करेगा एंड वो पीएचए का प्रोडक्शन जो है वो मिनिमाइज करेगा। सो व्हाट वी डू, वी प्ले विद these conditions. So the factors or the limitation or the limiting factors that actually help us, not the bacteria or any other microbe, is the nutrient limitation. अगर हम मीडिया में कार्बन का सोर्स बढ़ा के रखें बट फॉस्फोरस एंड नाइट्रोजन को लिमिट कर दें, तो these microbes has a tendency to produce more and more inclusions. Similarly, अगर हम उस किसी माइक्रोब के स्पेसिफिकली पीएचए प्रोड्यूसर्स के इन्वायरमेंट में ऑक्सीजन की मात्रा को कम कर दें, आप जानते होंगे कुछ स्ट्रिक्ट एनारोब्स होते हैं, कुछ फैकल्टेटिव एनारोब्स होते हैं और कुछ ऑब्लिगेट एनारोब्स होते हैं। अगर हमारा माइक्रोब एरोब है तो हम उसको एनारोबिक कंडीशन नहीं दे सकते क्योंकि वो सरवाइव नहीं कर पाएगा, पर हम उसको पार्शियल एनारोबिक कंडीशन में अगर ग्रो कराएं जहां पे ऑक्सीजन है बट सफिशिएंट मात्रा में नहीं है, तो in that condition also these microbes have tendency to produce more and more amount of pHs. There could be many other similar factors जो कि इन माइक्रोब्स में पीएचए प्रोडक्शन को enhance कर सकते हैं, and these vary from species to species. So this is how we try to regulate our environment in order to get more amount of PHA from the cells. These polymers, the PHA polymers, they have very similar structure, then and the very similar physiochemical properties with like plastics. These are biodegradable. They have high hydrophobicity because they are basically lipid granules, and their strength and flexibility is also very good. And that's why their application as bioplastic is very high. The accumulation of these PHA is triggered in the microbial cell when they are subjected to these physicochemical perturbance or the nutrient perturbance. Now let's talk something about the microbial PHA. अब इसके पहले हम माइक्रोबियल पीएचए के बारे में बात करें, I believe हम सबको कल्चर मीडिया के बारे में नॉलेज होगा, और अगर नहीं है देन वी नो मीडिया बेसिकली किसी भी माइक्रोब का खाना है जो उसको जरूरी न्यूट्रिएंट्स प्रोवाइड करता है ग्रोथ एंड मल्टीप्लिकेशन के लिए। नाउ जब हम लैब में सिंथेटिक मीडिया बनाते हैं यूजिंग न्यूट्रिएंट अगार एंड अदर कंपोनेंट्स, इट्स इजी टू ग्रो देम। बट अगर हमें अपनी टेक्नोलॉजी को इंडस्ट्रियल स्केल पे ले जाना है तो वी कैन नॉट वर्क विद दी सिंथेटिक मीडिया। वी नीड टू लुक फॉर सम substitute to these media, and that substitute has to be eco-friendly, should be cheap and economic, and at the same time should be renewable. So generally what we do, we look for all the industrial या agricultural waste. Industrial waste जैसे कि टैनरीज होती हैं, टैनरीज में लेदर की प्रोसेसिंग होती है, उनकी पॉलिशिंग होती है, और जब ये प्रोसेस खत्म हो जाती है तो वहां से जो वेस्ट निकलता है दैट हैज वेरी हाई लिपिड एंड कार्बोहाइड्रेट कंटेंट. Even the wastewater from the tanneries has high lipid and carbohydrate content. And similarly, other industries like wineries या फिर starch processing industries, या अगर हम एग्रीकल्चर की बात करें तो जब हम अपने सीड क्रॉप्स को हार्वेस्ट कर लेते हैं उसके बाद जो स्ट्रॉ बचा है, जो लिग्नोसेल्यूलोसिक बायोमास निकलता है किसी भी फील्ड से, जिसको जनरली भूसे की तरह यूज किया जाता है और फॉडर की तरह जानवरों को या तो खिलाया जाता है, और जब एक्सेस में होता है तो उसको बर्न कर दिया जाता है, जिससे कि पोल्यूशन बढ़ता है एंड प्रॉब्लम्स, इकोलॉजिकल इश्यूज बढ़ते हैं. So what we attempt is we attempt to use these kind of waste, whether they are industrial or agriculture. इन वेस्ट का हम प्री ट्रीटमेंट करते हैं थ्रू डिफरेंट प्रोसेसेस, एंजाइम डिग्रेडेशन, केमिकल हाइड्रोलिसिस के थ्रू, एंड उसके बाद जो हाइड्रोलाइजेट बचता है उसको हम यूज करते हैं एज अ फूड फॉर दीज माइक्रो ऑर्गेनिज्म. सो नाउ आवर मीडिया जिसमें हमें इन माइक्रो ऑर्गेनिजम्स को ग्रो करना है इज नॉट अ कॉस्टली सिंथेटिक मीडिया जो हम जनरली लैब में बनाते हैं, बट आवर मीडिया इज समथिंग विच इज वेस्ट फॉर एवरीवन, एंड सब उससे बेसिकली निजात पाना चाहते हैं कि ये वेस्ट किसी तरह किसी पॉजिटिव वे में कोई यूज कर ले, क्योंकि उसको फेंकना या जलाना या डिस्पोजल जो है वो ना एनवायरमेंट के लिए अच्छा है और ना ही वो पॉकेट फ्रेंडली है. तो हम उन वेस्ट को यूज करेंगे, उनको प्रोसेस करेंगे, उससे हाइड्रोलाइजेट बनाएंगे, उस हाइड्रोलाइजेट को हम एज अ मीडिया यूज करेंगे फॉर द ग्रोथ ऑफ दीज माइक्रोबियल कल्चर्स. The way it sounds, it takes time to develop and optimize such medias. एंड इस डेवलपमेंट एंड ऑप्टिमाइजेशन के लिए हमारे पास बहुत सारे प्लानिंग सॉफ्टवेयर्स होते हैं जिसमें हम अपने एक्सपेरिमेंट्स को डिजाइन करते हैं एंड प्लान करते हैं, एंड देन फाइनली वी कम अप टू एन कंप्लीटली ऑप्टिमाइज्ड कंडीशन दैट कैन बी यूज्ड एज एन ऑप्टिमल मीडिया फॉर एनी कल्चर. सो नाउ वी नो दैट हमारा जो मीडिया है इज बेसिकली a feedstock which is coming from waste from industries and agriculture. The next thing which is important for PHA production, अभी खाना तो हो गया, लेकिन अभी क्या बचा है? अभी बचा है हमारा culture. Now culture has to produce PHA, right? लेकिन PHA production को अगर बढ़ाना है तो अगर हम एक ऐसा culture select कर पाए जो कि पीएचए भी बना पाए एंड एट द सेम टाइम जो हमारा वेस्ट है, जैसे कि मान लीजिए हम एग्रीकल्चर वेस्ट की बात कर रहे हैं जिससे लिग्नोसेल्यूलोज आता है, तो वो लिग्नोसेल्यूलोज को भी हाइड्रोलाइज कर पाए. लिग्नोसेल्यूलोज को अगर हाइड्रोलिसिस होगा तो उसमें से सेल्यूलोज, हेमीसेल्यूलोज एंड लिग्निन निकलेगा, जिसमें से सेल्यूलोज एंड हेमिसेल्यूलोज इज द कार्बन फ्रैक्शन, जिसको बैक्टीरिया रेडिली यूज कर पाता है अपने ग्रोथ के लिए। तो अगर एक बैक्टीरिया ऐसा हो जो कि दोनों काम कर पाए, so that will be the best answer for the technology development. So इसके लिए हम क्या करते हैं? There are two ways. The first way is to make a genetically engineered microbial species, जिसमें हम अगर वो ऑलरेडी पीएचए प्रोड्यूसर है तो उसमें एक ऐसा जीन डाल दें जिससे कि वो हाइड्रोलिसिस भी कर पाए। एंड सेकेंड इज लुकिंग फॉर सम इंडिजिनस स्पीशीज जो ऑलरेडी ये काम ऐसी जगह कर सकती है। And the other way out is, rather than looking for one culture which can do multitasking, use multiple cultures. That means जब आप हाइड्रोलिसिस करना चाहते हैं तो आप एक कल्चर ऐसा लें जो कि हाइड्रोलिसिस कर पाए और दूसरा कल्चर ऐसा लें जो कि पीएचए प्रोड्यूस कर पाए। बट दोनों की ग्रोथ कंडीशंस सेम होनी चाहिए, दोनों की ऑप्टिमल कंडीशंस फॉर देयर typical activity should be same because we are going to add them together in the selected media. So यहाँ नीचे आप एग्जाम्पल में देख सकते हैं कि जैसे स्लॉटर हाउसेज या टैनरीज से या इंडस्ट्री से जो लिपिड वेस्ट आया है, उस लिपिड को हाइड्रोलाइज करने के लिए हमने सूडोमोनास ओलियोवेरेंस का यूज किया है, वाइल पीएचए प्रोडक्शन के लिए हमने क्यूप्रियाविडिस निकेटर का यूज किया है। एंड जब दोनों आपस में एक्ट करते हैं ऑन द सबस्ट्रेट, देन Cupravidus nicator is able to produce good amount of PHA in itself, while Pseudomonas species help Cupravidus to reach to the monomeric components of the feedstock by hydrolyzing the feedstock. One thing which is very important is to know about the indigenous strains. जब हम रिसर्च करते हैं, सबसे इजी तरीका होता है किसी स्ट्रेन पे काम करने का कि अगर आपको जेनेटिक इंजीनियरिंग वाला या मॉलिक्यूलर बायोलॉजी रिलेटेड काम करना है, यू गो टू सम माइक्रोबियल बैंक्स. Like India में we have MTCC microbial type culture collection in Chandigarh, and similarly worldwide ATCC American type culture collection in US. They are famous. तो अगर आपको जेनेटिक इंजीनियरिंग जैसा काम करना है जिसमें एक प्रॉपर स्ट्रेन के साथ वर्क करना है, then we try to get our strains from these culture collections. But when we are doing the work like this, where we need a dominant species जो कि पीएचए प्रोड्यूस करे या जो कि हाइड्रोलिसिस कर पाए वेस्ट का, we prefer using an indigenous species. अब indigenous species क्या है? Indigenous species or the cultures are the one जो आप अपने local environment से isolate करते हैं, जहां से आप अपना फीड स्टॉक ला रहे हैं या सिमिलर कंडीशंस जहां पे आप उसको ग्रो कर रहे हैं, आप वहां से कुछ माइक्रोब्स आइसोलेट करें, उनको उन कंडीशंस में डाल के चेक करें कि क्या वो ग्रो कर पा रहा है, एंड if it is growing well, then that is the most suitable strain for you. And it is always advisable for such work, go for indigenous strains. And they always respond very nicely to your experiments. Like when we started doing our experiment, we procured some cultures from CCAP UK. And at the same time, during performing the experiments, we observed कि कुछ growth naturally हो रही है हमारे हमारी plates पे. And then we observed that the growth which naturally happened because of the indigenous, these indigenous species, which we sometimes refer them as contaminants, but they also work well if they are degrading. So you need to go for a complete analysis of your of that culture later. But if it is responding the way it should be, you can definitely go for those indigenous culture. सिमिलरली हमने अपने लोकल रीजन से काफी सारे माइक्रोबियल आइसोलेट्स निकाले हैं एंड दीज आइसोलेट्स which we have isolated from extreme conditions even like Mariana Trench, we have observed that they are performing much better, they are adapting to these conditions in better way than the procured cultures which we have procured from some other country and have bring to our country. The genetic engineering of strain for enhanced PHA production and accumulation, one of the example is that the recombinant lipolytic strain of Delphia acidovirens. So this strain is originally known for its PHA production, but does not have a lipolytic activity. So what we have done, not me, some other researchers basically, what they have done is that they have added the gene responsible for the lipolytic activity into this strain. Now this strain, in addition to PHA production, can also do the lipase activity. So can also hydrolyze its feedstock, and after hydrolyzing can utilize that product itself as its food and can grow PHA granules into it. So these are some of the ways where we can screen some good PHA producers from our environment, or we can develop through recombinant DNA technology some promising recombinants that can help us in coming up with a suitable or complete technology for PHA production. Now there are variable factors that affects PHA production. These can be a combination of strains, its substrates, and what is the source of that substrates, the physical culture conditions, the variations in nutrient, and at what scale we are doing the process. The scale of process also plays a very key role, because when performing a process at flask level and then upscaling it to reactor level, you will always find a difference. The culture need to get adapted to or acclimatized to these conditions. Many times, or most of the times, the conditions in a reactor, they are more strict and they allow growth of exenic culture or the pure cultures more proficiently than at a flask scale.
Hello.
For the nutrient variation, as we have discussed, the variation in nutrient like high carbon content, low nitrogen, and low phosphorus content, it helps in achieving good numbers or good quality as well as quantity of PHA granules in a cell. Let's talk about some more PHA producers. So when we talk about microbial cultures that can produce PHA, there can be variety. Some of the majorly studied species are some bacterial strains, some actinomycetes, and some algal cultures. Algal culture is comparatively is a recent development, जिस पे अभी काम recently शुरू हुआ है, but the most used or exploited microbial cell for PHA production are the bacterial cells. Cupria viridis nicator is one of the most common bacterial cell which has been excessively used for PHA production. Many times we consider it as our reference strain when we start working with some new strain. The other bacterial strains can be Bacillus species, Azotobacter, Alcaligens, Methylotrops, Pseudomonas, and so on. The species with which our group is working is basically Actinomycetes, and they all are indigenous species, which my group has isolated from the local or niche area. And they majorly include Streptomycetes and Godonias and Micromonosporas. The algal cultures that has been explored till now majorly includes Spirulina platensis, Nostoc muscorum, and Sinococcus. This is quite interesting because algae are photosynthetic, and growing them will be more promising because they release oxygen to the environment. And many of these species are in addition to PHA, they are also rich in the protein content. So the valorization of the whole process will be much higher if it actually works at the larger scale. Many times, instead of working with one strain at a time, researchers prefer working with mixed cultures, like adding some algal strains and working together with some bacterial and algal strain, and or a mix of bacterial and Actinomycetes strains. They give better. They utilize the feedstock, the renewable feedstock, much more readily, and they produce PHA in a higher quantity. And these all combinations, they differ from species to species, and they highly depend on the nutrient condition of the environment and the physical condition of the environment. So as we know that the PHA are linear polyester produced and deposited as water insoluble amorphous and intracellular spherical inclusion or granules. These are generally 0.2 to 0.7 mm, and these are produced by a PHA metabolic cycle. Here I will introduce some of my cultures which we are working in the lab, although we have some very high amount of culture, good numbers of culture, but I'm just showing some representative cultures here. And as I have stated, only the Cupra vidus nicator, which is the first one, if you can see, is the bacterial culture, while rest of them are the Actinomycetes. The one which I have shown here, they all are very promising in PHA production, although they differ a lot in their morphology. These are the gram stain cultures, and you can see the Nicator and Gordonia BG1.3, they are negative, but rest of them looks like positive. And because they are indigenous strains, so they are the actually wide variety of bacteria and actinos with which we are working. It is difficult to state, to give a category in terms of gram staining for PHA production, because both gram negative as well as gram positive bacteria has reported to produce PHA in their cell with varying compositions. So it results in immensely diversified materialistic properties. And Cupravidus nicator, which is a gram-negative bacterium here, is the most studied culture for PHA production. When we analyze this amazing microbial world, it's important to quantify as well as to qualitate the PHA content in the cell of these microbes. For qualitative and quantitative both purposes, we can use confocal microscopy. Now, what is confocal microscopy? It is a technique that uses laser and fluorescence to create a three-dimensional image of a sample. A focused laser beam is used to excite the molecule at one point of the sample. The molecules are called fluorophore. Here we have used Nile red staining, and that's why you can see all the cultures which are stained here are in fluorescent red color. Although the color can vary from red to yellow, the Nile red is used to localize and quantitate lipids. So as we know, these inclusions are lipidicious in nature. So Nile red has a characteristic to localize the lipid which is there in the wherever it is present in the cell. So not to forget, the membranes of these inclusions are also of lipid nature. So they also get stained. And under the microscope, you can clearly see whether it's the membrane or it's the inclusion. In most polar solvents like water, Nile red fluorescence नहीं करेगा. But जब उसे लिपिड रिच एनवायरमेंट मिलेगा तो वो इंटेंशनली फ्लोरेस करेगा and the color, as I said, will vary from deep red to strong yellow or gold emission. So you can see all these representative microbes, the actinos and the bacteria, they look like producing good amount of PHA. We do have some other cultures which shows negative results because they didn't produce PHA in good quantity or quality. So this is the confocal microscopic qualitative analysis for the presence of PHA granule in the microbial cultures. The Micromonospora G3, you can see these were filamentous. So they have stained a little different because the granules are inside the filament in a thread-like structure. Then after that, to confirm the presence of PHA, we do STEM, which is scanning transmission electron microscopy. Scanning transmission electron microscopy is a hybrid electron microscopic technique which is used for imaging and morphological characterization with atomic scale resolution. इसमें आप देख सकते हैं कि अगर आप निकेटर को देखें, I don't know आप में से कितने समझ पा रहे हैं, but निकेटर में you will not find inclusion here. While Gordonia, all the three Gordonias, you can see a shiny spot. That spot is basically PHA inclusion, and that confirms the presence of PHA granules in these cells. Now you must be wondering कि अभी तक मैं आपको continuously बता रही हूँ कि सबसे ज्यादा studied culture is Cupravidus nicator और यहाँ पे nicator में ही नहीं है and rest में है. How is it possible? Because ये तो हमारा representative organism है. So not to forget, every culture एक specific stage of life cycle में ही granule banana shuru karta hai. When you are doing these microscopic analysis, at what day or at what stage you have harvested your cell and then you have freezed your cell in that stage is very important. सो मैंने यहाँ पे एक नेगेटिव रिजल्ट इसलिए लगाया है ताकि आप कंपेयर करके समझ पाए कि कहाँ इंक्लूजन है और कहाँ नहीं है. Like C indicator में on day one, the first day one, it will not produce the PHA granules, while rest of the but after day 3 it starts producing PHA granules. So the picture which you are observing right now, the first one for C indicator, it is from day 1, and that's why you cannot see any granule inside. But for especially the Gordonia BS2 and Gordonia JC51, you will find some good quantity, good number of granules inside that is actually covering most of the cell. It is at day 3 and day 4. Now, the production of PHA at a specific day and at a specific stage of life cycle varies from culture to culture. If for last two cultures it is giving granules at day 3, then there is a possibility that for BG 1.3 it will start producing granules at day 4 or day 5. And that's why whenever we do these kind of analysis, beforehand we need to have a good growth idea and an idea at what stage it is producing PHA. So for that, what we generally do, we have some chemical test which is known as crotonic acid assay, and it's an assay where we extract PHA, and finally after extraction, when it reacts with under acidic condition, it gives color, and that the intensity of that color when observed at 235 nanometer in a spectrophotometer gives you the quantity of PHA produced during that day of the growth cycle for that particular culture. So before going to the scanning electron microscopy or the Nile rest training, we prefer doing a quantitative analysis through those chemical assays so that हमें आइडिया हो जाए कि हमें किस स्टेज में इनकी स्कैनिंग इनका स्टेम एनालिसिस करना है या इनका कॉन्फोकल माइक्रोस्कोपी के थ्रू इनको व्यू करके इसको जस्टिफाई करना है. Now, PHA biosynthesis. So in the figure, you can see that a brief illustration of the mechanism that justify significance of nutrient depleted or limited conditions. The presence of ample amount of nutrient in the microbe, the Krebs cycle lead to production of high quantity of coenzyme A. तो जब तक मीडिया में बहुत ज्यादा न्यूट्रिएंट होगा, the carbon source and the vitamins and the nitrogen and the phosphorus, everything जब तक बहुत ज्यादा अमाउंट में होगा, तब तक सेल क्या करेगा, वो एम्पल अमाउंट वो क्रेब्स साइकिल में की तरफ लीड करेगा, क्योंकि वो बहुत हाई क्वांटिटी में कोएंजाइम ए का प्रोडक्शन करेगा. एंड जब तक कोएंजाइम ए हाई क्वांटिटी में प्रोड्यूस होगा, यू कैन सी द पीएचए एंजाइम ये ऑब्लिटरेट हो जाएगा, इसकी एक्टिविटी को वो स्टॉप करेगा. PHA enzyme is also known as 3-ketothiolase, and if the activity of this is blocked, then the accumulation of PHA will also be blocked. तो अगर हमने न्यूट्रिएंट्स बहुत हाई अमाउंट में डाल दिए हैं, तो क्योंकि क्रेब्स साइकिल की तरफ वो डाइवर्ट होगा विद एसिटिल कोएंजाइम ए प्रोडक्शन, तो PHA एंजाइम जो है उसकी फंक्शनैलिटी ब्लॉक हो जाएगी, and it is not going to go to further produce or accumulate PHA in the cell. बट अगर न्यूट्रिएंट लिमिटेड कंडीशंस हैं तो PHA जीन जो है ये A जीन एक्टिव रहेगा and this will direct the flux of acetyl coenzyme A towards the PHA production. The successive action of these three prime enzymes, which is PHA-A, PHA-B, and PHA-C, is responsible for the three essential reactions. PHA-A will eventuate the reaction between two acetyl coenzyme A that will lead to the production of acetyl coenzyme A, and subsequently PHA-B जो कि एक NADPH dependent acetyl-coenzyme A dehydrogenase है, वो acetyl coenzyme A को further reduce करेगा into 3-hydroxybutyryl coenzyme A. 3-hydroxybutyryl coenzyme A मोनोमर है एक पॉलीहाइड्रोक्सी एल्केनोइड का. फाइनली जो हमारा लास्ट एंजाइम है विच इज PHA-C, वो इन सारे मोनोमर्स को ज्वाइन करेगा, will lead to the polymerization of these monomers, and that will give polyhydroxybutyrate, which is a kind of polyhydroxyalkanoid. So this is how the microbial synthesis of the PHAs are triggered at genetic level. And further, once the PHA is produced, its application is definitely in all the fields you can think of: biomedical, environmental, industrial, and the best properties that makes it suitable for our uses is that they are biodegradable, they are biocompatible, and at the same time, they are thermostable. Talking a little about biodegradation of bioplastics. So, अगर हम first blocks देखें अपने, जिसमें synthetic polymer है, that degrades in lot and lots of air. It degrades into fragmented polymers or the oligomeric chains, and these chains are basically the small oligomeric chains. They lead to the production of microplastics. We have already discussed that these microplastics, they create lots of environmental issues, and they enter the food chain of the organisms, and we really don't want them. But when we go for the biodegradation route, then after the biodegradation, the water, carbon dioxide or methane is released from the microbial biomass, and the sludge which is remain can further be used as a manure, and these can come to the environmental cycle and can be recycled easily. The various microbes that can assist in biodegradation can be aerobes, anaerobes, photosynthetic bacteria, archaebacteria, eukaryotes, and fungi, etc. They generally secrete intracellular or extracellular enzymes. Some of the PHP, which is polyhydroxybutyrate degrader, are depolymerases, which originates from rhodospirine rulum, while PCl degraders are depolymerases from Streptomyces thermovivacius. Lipases that which are from Alcaligens fecalis, and esterases from Commonas acidovirens. Certain biopolymers like PBS, they can be biodegraded with very efficiently when with the joint effect or the co-culturing of Fusarium solani and Stenotrophomonas maltophilia, while the PCl degraders that effectively degrades PCL are the Streptomyces species and the Bacillus. Biodegradation in the environment sounds like a waste treatment method. The term biodegradation refers to the degradation performed by naturally occurring bacteria in the nature, while the compostability define complete degradation of material into water, methane, and under the control condition. 90 से ज़्यादा microorganisms are capable of doing these kind of biodegradation of bioplastics. These microorganisms are extensively found in soil. They are mostly soil borne, and they are very easily can be found or extracted from any compost material. As we have seen that the co-culturing of different microorganisms may enhance biodegradation of bioplastic, and in fact the other microorganism that helps in biodegradation by utilizing the intermediate bioplastic is by the main organism. So it's always like a group work is better than a single-handed task. So coming back to the PHA cycle, I think now this will be more clear to everyone that we are taking waste, whether it is agricultural or the industrial waste, and trying to utilize those waste by various microbes like bacteria and actinose and even alga. They undergo fermentation, and the cultures are then harvested. The harvested cultures, from those harvested culture, we extract PHA, and from those PHA, we produce biodegradable plastic. These plastics, when their use is finished, they can be collected from the waste. They can go to the recycling units and can again come up as a fresh, as a new bioplastic material. Or else, if they need disposal, then they can enter into the anaerobic digesters. These plants, the anaerobic digestion plants, will digest them, and in turn the waste which is generated from these plants can be used as biofertilizer or manure, which can assist again in the agricultural field for growing their crops efficiently. So this is how the whole process contributes to the circular economy in the most eco-friendly way. Definitely, the whole process is very promising and sounds it should work well, and it does work well, but the challenges are always there. Some of the challenges are to find a dominant, suitable strain that can work efficiently for your feedstock. Screening of those cultures and selecting those cultures that can effectively not only hydrolyze the feedstock anyone is looking for, at the same time should also be able to produce good amount of PHA. The most important challenge is the high production cost, and this high production cost generally comes from the price of the feedstock. That's why we are focusing on the alternate feedstock that are not pricey, but at the same time, when we look for these feedstock, we need continuous supply of these feedstock throughout the year, because we cannot run an industry where a feedstock is available for 2 or 3 months, and then you need to shut down the industry for 3 months and wait for another harvest, and then you can again start. The industry has to go on, so the continuous supply of feedstock is important. And once everything is scaled up at the laboratory scale, you need to upscale it at the industrial scale, and it should work in the same promising manner at that scale. And these are the few challenges which we generally face when we want to bring our technology on field from lab. So some alternate substrates which we all have been working with is the agricultural waste, the lignocellulosic biomass we have already discussed about, the tannery waste, some microalgal cultures, some crude glycerol as which is waste from the industry, some vegetable oil, some dairy waste, some winery waste. Vegetable oil is not a good option because we all know we come from developing countries, and although the feedstock like corn or the vegetable oil, they actually work very nicely, but we cannot use them for the feed and food fight. So we need to focus more on the waste utilization and valorization of waste rather than looking for something which the general public needs from day-to-day life. Finally, application of PHA. To be frank, आप प्लास्टिक के बारे में जब भी सोचते हैं शायद ही कोई ऐसी जगह होगी जहां पे उसका यूज ना हो. It's literally everywhere. And because the properties of bioplastics are very much similar to the conventional synthetic plastics, they can be used everywhere where a normal conventional plastic is used, whether it is for the packaging of food or it is for the for the medical utility or for the agriculture applications for making any tool or as a wood composite for making some furnitures. It's literally everywhere. So the application, generally we select the application only after we characterize the PHA or the bioplastic we have recovered, and after knowing its tensile strength, how brittle it is, how flexible it is. After having complete characterization for our extracted bioplastic, we decide that where my set of extracted bioplastic can be utilized further. So in conclusion, I can say that the microbial bioplastics are potential substitute for petroplastics. A huge diverse group of microbes can be studied for the synthesis of these bioplastics. The most interesting part is under optimal conditions for PHA production, a microbe can accumulate as much as 80 to 90% of PHA. And the best part is the attempt to utilize the unconventional feedstock, which is mostly waste, can support the economy of the whole process. And the other way to support the process is the simultaneously degradation of feedstock and PHA production, if possible, by finding a dominant strain or by co-culturing or by mixed culture or even by developing a recombinant strain that can do multitasking. The dominant PHA producer with hydrolyzing ability, these can be definitely produced through genetic engineering, and depending upon the properties, the application of bioplastics are widespread, and it is nearly in all the fields which we can think of. So thank you so much for listening to me. I hope you get something good from here, and I don't know. I can't. Unfortunately, I can't hear you. Also, I'm unable to listen the responses.
Thank you, ma'am. Thank you so much for your nice talk because it is actually extremely needed that we use less petroplastic, which is actually not biodegradable and create environmental issues. So definitely, the lecture is very informative, ma'am. Thank you so much. Pratima, next participant is Pratima.
Sir, can I present my e-poster presentation?
Pratima, you can start.
Yes, sir. I am audible.
Yes, audible.
Okay, thank you, sir. Yeah, 2 minutes support करो बाहर से A very good afternoon to all of you. Myself, Pratima Shrivastava. I am research scholar in Department of Biotechnology from Veer Bahadur Singh Purvanchal University, Jaunpur. Today, one of the major challenge in front of world is emerging infection and resistance of pathogen against existing antibiotic. In this
अरे
context,
मुझे तो कुछ सुनाई नहीं दे रहा था, मैं तो हवा में प्रेजेंटेशन दे रही थी.
hello, hello, I am audible. Hello, hello, I am audible.
Yes, you are audible. You can continue.
Okay. Thank you, sir. Today, one of the major problem in front of world is emerging infection and resistance of pathogen against existing antibiotic. In this context, scientific community have to focus for obtaining various metabolites with wide spectrum bioactivity. I am working on the bacterial endophyte, so my presentation topic is evaluation of antimicrobial and plant growth promoting trait of bacterial endophyte for turmeric. The term endophyte, first time coined by D. Barry, endophytic bacteria can be defined as those bacteria that colonize to the internal tissue of plant and causing no sign of infection. Endophytes live in each and every parts of the plant. These are the various applications of endophytic bacteria. Endophytes act as excellent source of antibiotic, antiviral, and immunosuppressive compound. Endophytes have ability to promote the growth of plant. Endophytes have improved the plant health by protecting from the pathogen. Endophyte also used to control the pollution. I choose the turmeric plant for isolation of endophyte because turmeric is known for its medicinal property. These are the various medicinal property of the turmeric. These are the objective of my work: isolation of bacterial endophyte from rhizome of turmeric, in vitro efficiency assessment of plant growth promoting trait of isolated endophyte, and screening of antifungal activity of the bacterial endophyte. Samples were collected from a turmeric grown field of Jaunpur, and isolation of endophyte were done by using surface sterilization method, determination of plant growth promoting trait. These are the various PGBR traits that determined by using these methods. For isolation of endophyte, first healthy rhizome were collected from the field and surface sterilized by using 70% ethanol, 0.5% sodium hypochlorite for 3 minutes, and finally treated with the 70% ethanol for 30 seconds. To check the effectiveness of the surface sterilization process, last was spread on the nutrient agar plate and incubated for 48 to 72 hours. If no growth in control plate, then isolated bacteria are the endophyte. These are result of the IAA production. Among all the isolate, SS05, 7, and 11 show the maximum IAA production, and SS02 show the minimum IAA production in presence of tryptophan, whereas SS05 and SS04 show the maximum IAA production in absence of tryptophan. These are result of ammonia production. Among all the isolate, 84% of the bacteria have ability to produce the ammonia. These are the result of phosphate solubilization. Phosphate solubilization were determined by using NBRIP media. Among all the isolate, SS07 show the maximum solubilization of the phosphate. Potassium solubilization were done by using Alexandro media. Among all the isolate,
आवाज क्लियर नहीं आ रही, थोड़ा आवाज क्लियर करिए.
आवाज आ रही है सर? हेलो.
नो मैम, हेलो, आवाज आ रही है, आप बोलें प्लीज.
Okay, thank you. SS05 show the maximum potassium solubilization. Zinc solubilization were done by using modified Picovskaya media. Among all the isolate, SS05 show the maximum solubilization of the zinc. Siderophore production were done by using chrome azurol assay, and presence of yellow hollow clear zone around the colony show the positive result of siderophore production. Among all the isolate, SS05 show the maximum production of the siderophore. ASEAN production were done by using King's B media supplemented with the 4.4 gram glycine per liter. Among all the isolates, 16% of the bacteria have ability to produce to the ASEAN. When bacteria have ability to produce the ASEAN, then ASEAN reacts with the filter paper and change the color of filter paper from yellow to dark brown. Antifungal activity against Fusarium oxysporum among all the isolate SS05 show the maximum antifungal activity and inhibit more than 70% of the mycelial growth of the Fusarium oxysporum. Biocontrol activity against Fusarium odor among all the isolate tested, SS07 show the maximum antifungal activity and inhibit more than 60% mycelia growth. And biocontrol activity against Rhizoctonia solani, SS07 show the maximum antifungal activity. Finally, bacterial endophyte, they are isolated and characterized for plant growth promoting activity. The result often suggested that SS05, 7, 17, and 20 exhibit multiple growth activities and antifungal activity against Fusarium odum, oxysporum, and Rhizoctonia. The plant growth promotion activity should be confirmed after field trial, then final conclusion to be drawn. The isolated endophyte may be used as a biofertilizer and biocontrol agent. Endophyte having antimicrobial activity will be useful for searching some novel compound that prevent the emerging infection and drug resistance. Thank you. First of all, I would like to thank Department of Science and Technology for providing me financial assistance under DST Women Scientist A scheme. I would also like to thank Professor Rajesh Sharma and Dr. S.P. Tiwari for providing lab facility and support. Thank you.
Okay, thank you, ma'am.
Sir, can I present my e-poster presentation?